Hi. Welcome back, everyone. Had a really good smoker here. Famous sausage rolls from the Coonawarra store. Good way to warm up. It's a nice day here. We've had a bit of rain already, a bit of sun. We'll probably get the same again a little bit. But on with the program. So the next speaker is from New Zealand. Welcome, Vaughan. I'll just read you his bio, and then we'll go cross over to him. So Dr. Vaughan Bell, Senior Scientist, Wine Grape Entomology and Virology, part of Pest Management Science New Zealand Institute for Plant and Food Research. Vaughan Bell is a Senior Scientist working for the New Zealand Institute for Plant and Food Research, Havelock North. Since 2004, he has studied sap-feeding mealybugs in vineyards with an emphasis on their ecology, biology, and ability to transmit or vector grapevine viruses. By examining the interrelationship between the vine and virus vector, his findings have contributed to the development of a practical and financially sustainable virus management response commonly deployed in commercial vineyards in New Zealand. Over to you, Vaughan.
Thank you, Ulrich. Can you see my screen okay? Yes? Really hard doing these webinars because I can't eyeball the audience there, so I'm going to have to assume that everyone can see that, and hopefully you can hear me clearly. I've changed the title just slightly. I've added the word possible because being a Kiwi, I don't want to appear too arrogant to suggest that what we're going to propose or what I'm going to propose is a template for Australia. It's a possible template. There are some caveats here, so I'll talk about those in a second. But there are a number of colleagues here, too many to list on the screen, but I would like to shout out to them for everything they've done with this presentation. Okay, can you hear me okay? This presentation today is it's really going to provide you with some steps along what is a path that's been well trodden in New Zealand now for probably at least the last 12 years, possibly dating back even a little further. I'm going to highlight to you really what we call an integrated response to leaf roll virus. You'll hear me use that word integrated a lot. It's a multi-tactic response. It relies on no single factor on its own. It's a range of tactics that actually control this, that help manage this disease. But importantly, they're practical, and they can be implemented in the vineyard. I'm going to offer some considerations for Australia towards the end of the presentation. And on the left there, we have the long-tailed mealybug, one of the mealybug species found here in New Zealand. And in the middle, I added that more recently because, as Fiona highlighted in her presentation, scale insects are an issue for you, not so for us here in New Zealand. I'll talk a little bit more about that in a moment. This is the virus, the pathogen we're dealing with here, grapevine leaf roll associated virus 3. Fiona gave a wonderful overview of this pathogen earlier on. We too have leaf roll 1, and we probably also have Shiraz disease, a mix of leaf roll 3 and GVA. But this is the one that in our testing that we've done in the laboratory, we detect most commonly. And you can see in the red berry cultivars, the foliar symptoms are really quite distinct and are an important aid to managing this disease in our vineyards, which I'll talk about shortly. But I do wish to just highlight that for those of you who are growing whiteberry cultivars, the issue is more problematic because these vines can be infected with the disease, but they show no obvious foliar symptoms. Rootstocks are the same. So for a country like New Zealand, which as many of you will appreciate is dominated by Sauvignon Blanc, this is the elephant in the room, and there are at this point in time no economically feasible ways of managing leaf roll 3 or leaf roll virus in our whiteberry cultivars, and that will be an issue that you will find in Australia as well. So the focus of this presentation really is on managing the disease practically in redberry cultivars. New Zealand's response to managing leaf roll 3 really commenced in probably the late 90s, early 2000s, with the development of our propagation standards known as the New Zealand Wine Growers Grafted Grapevine Standard. There's been a number of iterations of that over the year. It's an incredibly important document. It is the foundation upon which really everything rests for virus management in New Zealand vineyards, and Fiona highlighted that. That is, it's fundamental to your efforts to manage viral pathogens in Australia as well. In 2005, the wine industry commissioned a large nationwide survey of growers to gauge their awareness and understanding of not only leaf roll virus, but also how to manage the disease, the insect vectors that were transmitting the disease, what was the awareness of those like, and how were people controlling both the vector and the virus. That was followed a year later by a literature review where we were commissioned by New Zealand wine growers to look at the virus, how to manage the virus, the vectors, monitoring and management of those. Then a couple of years later, we had the Virus Elimination Project, which was a 6-year project that commenced in 2009 that really focused on a particular growing region here in New Zealand. And we set about trying to implement a management response to this virus in the vineyard with the aid of the growers. So while there was a regional perspective here, so we looked at the entire 1,000 hectares roughly, about 24 separate wineries were engaged in that project. We also looked at a very fine scale at about 13 individual vineyard blocks within a range of those wineries. Each one of them focused on a single redberry cultivar, and we set about trying to we collect data from those and then share the learnings of that with the industry. And then, of course, since that concluded in 2015, we've set about on a number of different research projects around using ground cover plants as a potential trap for mealybugs. We've looked at other non-vetus plants as potential reservoirs for the virus. We've looked at mealybug insecticide improvement, how to get the most out of that technology, biological control using that natural ecosystem service, and of course mealybug pheromones as well as being part of that research program. Because the grafted grapevine standard and your equivalent of that in Australia is the bedrock upon which everything rests around virus management, I'm starting with this in terms of our integrated response. Here in New Zealand, we have 7 VENA members, varying sizes, variable penetration into the industry. All of them have a particular set of standards that they need to abide by in order to control the quality of the material that's disseminated to the industry. No one gives a guarantee that it is free, in our case of leaf roll 3, which is what they're screening for, but they provide an assurance. So they follow a set of very carefully designed, carefully set out and regularly audited protocols to ensure that, to the best of their ability, without, testing every single piece of material that passes through any one nursery, that they have got clean material. And that greatly reduces the risk of that material being distributed into the vineyards as they purchase material. So the Virus Elimination Project was an in-vineyard response. In our case, it focused on redberry cultivars. The objective was to develop and then test a practical, multi-tactic, integrated response to the disease to reduce the incidence to less than 1%. As I indicated, we focused on 13 individual study vineyards. They ranged in size from as small as 1/2 hectare to 2 hectares in size. And every year we'd go through, we would collect data related to the virus incidents, the patterns of spread that we saw of that disease in those vineyards. And every year we'd also do an assessment just before harvest to get an idea of what the background population of the mealybug abundance was like in the grapevines. So we wanted to understand what were the links between the numbers of insects, mealybugs in our case, that we would see in the vines, and how did that influence the virus management outcomes. And Fiona indicated about vector abundance being very important. It really, really is, as you'll see from the presentation later on. We wanted to know if we could reliably use those visual foliar symptoms that were highlighted by both Fiona and in earlier slides here to detect leaf roll virus and inform the vine removal protocols that would follow. So we wanted to know, is roguing, the removal of those individual virus-infected vines, is that a tactic that allows a vineyard owner to get on top of the problem, to manage the disease effectively, or do we need to look at some variation to that tactic, which I'll explain shortly. So we set about collecting data, we reported the results to the industry, and then throughout that 6-7 years of that program, there was an extensive extension program to industry, sharing our initial interim recommendations. And as our confidence grew, as the results started to kind of bed themselves in, we then moved from sort of interim results to confirmed results, which is where we're at in many respects for virus management today here in New Zealand. Typically, I've left the insect vectors to further on in this presentation, but the more I understand this disease, the more I realise actually they are really a pivotal part of this disease pathosystem, if you like. I've highlighted there, do not underestimate the role of the vectors, whether it's mealybugs or scale insects. They are absolutely fundamental to your ability to get on top of this issue. Remember, it's a vine virus vector interaction here. So it's actually a really complex and a really dynamic set of circumstances that are presented within your vineyards. So reducing vector presence in the vines is absolutely critical to managing the disease, managing leaf roll 3 effectively in your vineyards. And then, of course, in the vineyard itself, did I skip one? I did too. Insect vector management. Sorry about that. The default position here in New Zealand for many of our conventionally grown vineyards is to adopt an insecticide program. And the bedrock of that is primarily around 2 active ingredients, buprofizin, the product that you might know over there best, and that active ingredient is applaud. And of course, spirotetramat, which is a systemic product, Movento. It's a systemic product applied to the foliage and translocated through the vines. Buprofizin, of course, applaud is a contact. So it relies on hitting juvenile life stages of mealybugs or scale insects and preventing them from molting, leading to their eventual death. For the contact insecticides in particular, cover is king. We've got a spray specialist here in New Zealand, and his favourite saying is coverage is king. You've got to get good coverage of the vine canopy to get really good control of these these insects, which are often hidden in the old wood, in the cracks and crevices of that old wood, and of course under the flaking bark. In New Zealand, as of just this year, there are no longer organophosphates available to New Zealand growers. The last of them were removed from the spray schedule approved by the New Zealand industry as of the first pruning cut of this autumn 2021. They are for a lot of people, they were kind of the go-to tactic where they had really severe outbreaks of mealybugs, but they're highly disruptive. They will not only take out, some of the mealybug populations, probably not all of them, but they'll take out many of your natural enemies, the little predators and parasitoids that you might rely on in your vineyards to manage mealybug and scale insects. And on that note, biological control, I won't dwell on that here today, but it is a critical part of managing insect vectors, mealybug and scale insects. It's largely unseen, it's largely undervalued, but it will be happening. And if you apply the right tactics, the right products that are compatible with integrated pest management, you can really get some really good advantages from biological control in your vineyard, as studies here in New Zealand have shown. Visual symptom identification, I shorten that to VSI. Fiona made reference to this earlier on. On the left there, we have the classical foliar symptoms of leaf roll 3 and Pinot Noir, but there are a range of other often benign conditions that can give rise to foliar symptoms that look remarkably similar to those of leaf roll 3. And I'm aware of examples here in New Zealand where growers have gone through tagging vines, showing the foliar symptoms on the right there, which is magnesium deficiency, thinking it is leaf roll 3. Fortunately, they didn't pull those vines out, but it's, of course, the vines that I don't see that are lost that, could well be the untold story. But this is not a trivial task. It requires a lot of training and some expertise, but it does work. It really is a good way of being able to reliably detect leaf roll 3, leaf roll 1, and Shiraz disease in your vineyards in Australia. Certainly for us, it's the go-to product, and we went to that in lieu of laboratory testing because that comes with a cost. Not only a financial cost, but also in terms of delays for growers. There's a lot of logistical issues with collecting and labeling and then dispatching material to labs. So they needed something they could practically rely on in their vineyard to help them get on top of this problem. So part of the virus elimination project was determining whether we could use VSI as a means to identify those individual finds to inform roguing decisions that would follow in the months to come. And on that note, we got on to the subject of roguing. So this is the next stage. So once you've visually identified your vines and you've tied some flagging tape around the trunk, the vineyard team will typically, in New Zealand anyway, they're going through in June, July, and they're starting to rogue or remove those individual infected vines that had shown foliar symptoms in generally here in New Zealand is sort of March, April. Fiona indicated that the agreed threshold here in New Zealand for roguing is up to 20% incidence. There's a little bit of flexibility on that. It'll depend a little bit from one winery to another, but I think the industry standard is that anything above 20% virus incidence, it becomes really hard to economically justify continuing a roguing program. And that's particularly true if there's not good vector control. So anything up to 20% is, if you've got good vector control, roguing can and does work. We've shown that with real examples here in New Zealand. I hasten to add, though, that roguing will become an annual part of the vineyard response. It just becomes one of those tasks that you add in your vineyard to the work schedule. It's here, it is now at the new normal in New Zealand. Remnant roots are definitely an issue. We know from work we've done here that they are quite a long-term reservoir for the virus. And we do, on occasion, find at least one of our mealybug species, as you can see on the bottom right there, residing on the roots of grapevines and other host plants for that matter. The thinking is that if they're residing on the remnant roots of a virus-infected vine that is a reservoir for the disease, and newly planted vines are infected, there is a possible virus transmission pathway when the mealybugs move from the old remnant roots to the newly developing roots of the newly planted vine. So if that does occur, and we have, that's an intuitive, it's a hunch that that is occurring, but it kind of makes intuitive sense that that could be occurring. That is another means by which the disease may spread within our vineyards, because we do see new infections popping up in some newly planted vines from time to time. And they've come from nurseries that are part of the New Zealand Wine Growers Grafted Grapevine Standard. So, you know, has it come from remnant roots? We just don't know, but it's a possibility. We did some work recently looking at the economics of leaf roll virus here, and I want to have a shout out to my colleague here. This is Dr. Alistair Hall, who's recently retired from Plant and Food. It was a piece of work funded by New Zealand Wine Growers and a government department, Ministry of Business, Innovation and Employment. And effectively, we set out to take these real data that we collected over during the course of this virus elimination project and model those data in a simulated 1 hectare Merlot block containing 2,500 vines. And Alistair used his mathematical genius to take these data and come up with some really quite powerful insights into managing this disease in New Zealand. I'm not going to go into a lot of detail on that, but I want to take out, cherry pick if you will, a few key messages. Here are a couple of graphs looking at an initial virus incidence at the start of what was a 20-year lifetime of this simulation, this model simulation we did, 0.4 incidence disease and low numbers of vectors. And the definition for low, as per this model, was 6 mealybugs found per 100 leaves inspected. And you can see there's not a great deal of difference there between the roguing option, which flatlined at roughly an average of just under 1% per of loss of vines per annum over 20 years, versus the no action decision. So that's where a grower deliberately decides, I'm going to do nothing to manage the virus by way of visual symptom identification or roguing. So they just live with low numbers of mealybugs. And you can see over about 20 years, they get to about 8-9% cumulative virus within that block. Look to the graph on the right. At an initial 20% virus incidence, which is our roguing threshold, again, with low numbers of vectors, roguing successfully contained that to less than 1% over the 20-year life of the model. But with a no action response, the incidence of the disease after about 20 years increased to about 40%. Compare that with the same low incidence, 0.4, and the high, 20%, against a situation where you've got high numbers of vectors in your vineyard. And by that, our definition for this model, based on real data, was 75 mealybugs per 100 leaves inspected. And look at the slope of that black line in particular, both at the low incidence and at the high incidence of 20%. After about 13 years with no action, we had about 90% of the vines showed foliar symptoms of leaf roll virus. At 20% incidence at the start, that 90% of the vines were infected after just 8 years. The cost per hectare per annum for a roguing option worked out to around $131, as I say, per hectare per year. I'm just going to a note here. That was the sustained the lowest annual virus incidence, the least need to replace to plant replacement vines, and provided the lowest estimated average annual costs plus loss of income. So that reiterated the current recommendation in New Zealand to rogue the virus-infected vines, those vines showing foliar symptoms. Contrast that with the situation where you rogue at high vector density. The increase was around $5,400 per hectare per year. And that was basically made up of three main components. That was the cost of roguing itself, the yield loss from replacement vines over the course of that 20 years, and of course the yield loss from infected vines as well, because that is one of the symptoms of this disease. It reduces yield. So we've got a situation here, we've got a really expensive disease, and managing the vectors really matters. Roguing is important compared with the no action decision, but managing those vectors has a huge impact, not only in terms of the overall loss of vines through the course of that 20 years, but also as a financial, there's a financial hit. So I'm going to conclude here by providing a few ideas of what Australia's virus management template must look like. Good nursery propagation standards must be the baseline for those contemplating rogue and replant. It's there to screen for these viruses before you plant them in your vine. It really is incredibly, really important. With training, visual symptom identification should also be effective in Australia, but I think there are some really important variables that need to be considered there. The timing at which those virus symptoms might appear in Australia might be quite different to Australia, and that'll be based on factors like temperature. That may change both the extent of symptom development and the timing of that symptom development relative to New Zealand. So I think you need to be focusing on, how does VSI work? And of course, you've got so many more red berry cultivars that you grow over there compared with New Zealand. So some of those may have some very subtle differences that you might need to consider as you embark on a VSI program. Roguing works. It really does. We've got good evidence here in New Zealand to show that it does work, but it only works when it's supported by low numbers of insect vectors in the vines. You need to consider what your roguing threshold might be in Australia. Is it 20%? Might it be lower than that? These are things that you need to work out. Is roguing symptomatic vines the best course of action for you? Or might you have to look at perhaps roguing the within row vines either side of that symptomatic vine to get on top of this problem? These, again, questions that need to be considered by your Wine Australia and the various regional groups there and individual growers. Know your vectors, understand how their abundance in the vines influences virus management outcomes in Australia. This is really, really important. You want to minimise as much as possible the risk of virus populations overlapping with vector populations. What we found in New Zealand is if there's no virus, there's actually some tolerance for a few mealybugs in our vines. We don't need to eliminate them to actually, manage the issues around leaf roll virus. It's where you've got virus vector populations potentially overlapping and interacting that we see we've got the recipe for spread for this disease. Insecticides, they may be a necessity. That's something that has really been accepted here. There are a lot of people who didn't want to go down the insecticide route for some years to get on top of a vector problem while you're also reducing the virus incidence in a block. It may be something that you have to adopt as a necessity. Consider the product range that you've got available for mealybugs and scale insects. You want to look at availability and the compatibility of those products, the compatibility with biological control. And again, the timing of those products' use, the product rate and the coverage are really important factors to consider. And I guess my final comment would be that virus management, as we've found here in New Zealand, it's not easy. Adopt best practice at all times. Speak to people, ask questions, talk to your regional authorities about it and persevere. But one thing I will say with absolute certainty is that inaction is really not a viable option. You really do need to tackle this pathogen and tackle it as soon as you possibly can. Thank you, everyone, for this. Thank you to Kerry and Richard, Wine Australia, Limestone Coast and Wines of Western Australia. And of course, my New Zealand wine growers here and the government departments who have helped us along the way. Thank you very much.
Thank you, Vaughan. Great, and a lot of questions I think will come from that. Now, speaking of questions, just a reminder that we will, if you could keep them in mind, write them down, get them to Hans here, and also to Richard in Western Australia, and then at the Q&A, we can really go through them in some depth and detail. Thanks, Vaughan. Now for our first Coonawarra presenter, Kerry DeGaris. I'll read you out her bio, and she'll come and stand next to me, and then we'll set her up. So Kerry DeGaris. Dr. Kerry DeGaris, a viticulturist at Treasury Wine Estates, currently working part-time for Treasury as a viticulturist, providing technical support for company and grower vineyards across Australia. She has worked in the wine industry for close to 25 years. When not at TWE, she can be found at her family property at Bull Lagoon, tending to sheep and cattle and lambs. Lambs at the moment. Great. So welcome, Kerry. We'll just bear with us a minute while we set up to get the PowerPoint together.
Hello to everybody in Margaret River. I hope you have had some decent rain over the last few days. We certainly have, and we tend to get your weather a few days later. So hopefully, I think you guys have had more rain than we've had. So hopefully we're all looking forward to a nice wet winter. Okay. So I'm going to give a quick presentation to talk to you today around virus identification. You've seen lots of pictures already about virus, but I've brought it back to a Limestone Coast perspective. This talk is Kath Kidman is really meant to be giving these presentations today. I don't claim to be the virus guru, but I've certainly helped Kath out over the last 3 years now walking vineyards. I think last total we were up around the 820 hectares that we've walked across our company vineyards in South Australia this year. We average around that 12 to 15 Ks a day, just walking vineyards, as Vaughan has already mentioned, that visual symptom identification, which as our company has sort of taken the track that Vaughan has, that's the way that we're trying to tackle our virus problem. But to give you an idea of what we're seeing, so when we go out to do visual symptom identification, really we come across a red vine, which I'm imagining the people I'm looking at in the room would all know what I'm looking about this year, particularly for the Limestone Coast, was a very symptomatic year for virus. So I think we'd all be very conscious of when we walk out into vineyards, there's been a lot of red. Are we dealing with a nutrient deficiency or virus, or is it something like that photo there that you can see? You can see the damage. I'm assuming I can use this here. Can you see that damage there on that particular trunk there? And you see the red leaves. I mean, that's what takes you to that vine straight away. But of course, we wouldn't tag that vine out. We'd know that that's causing that redness is something else. So just to be aware, there's lots of other issues we need to be thinking about when we go out and do this visual symptom identification. As the previous speakers have already alluded to, we have varietal differences, and I'll show a few photos of that. Seasonal effects, I don't know what other people thought, but in this region, and I'd be interested to hear what Margaret River has to say about this, but this season was particularly bad for symptom diseases. My next talk that I give you, I'll put some pictures up of a more regional scale to give you an idea of the redness that we've seen in our region and how concerning it is. But is that attributed to the fact we were carrying very large crops this year, as well as we didn't have a very hot season, so we did get some cool weather earlier on? Are those two factors combining to produce a season where we're just seeing a more stressed vine, so therefore the redness is coming out earlier? I'm putting it out there. I don't claim to know the answer to it, but if you compare it to the previous season where we really didn't have any fruit at all on our vines, and we had a much warmer season, we just didn't see that symptomatic expression as much as we have in previous years. And once again, mechanical damage. And because a lot of the vineyards that we're walking young vineyards. So we're talking less than predominantly 5 years of age. And so mechanical damage is something that we see very commonly because just with vine training issues. So, and you know, kangaroo damage, seen a lot of that this year, and rabbit damage, those sorts of things, pests coming into your vineyard, you'll often get red leaves as a result of that. Right, so knowing that we had to do this talk, it's always good to take photos out in the vineyard and make sure that you then test those vines to make sure you can get some positive test and prove what we're talking about. So I just wanted to emphasise, we talked a lot about leaf roll 1 and leaf roll 3 are probably the two main leaf rolls that we see in this region that we consider to be a problem. So if you look at your right-hand side, that is a typical leaf roll 1, leaf roll 9 GVA vine Cabernet. I'll emphasise we mainly look at Cabernet, as you can appreciate, in this region. To the left, though, is positive for leaf roll 3, leaf roll 9 and GVA. So really the difference there is the one on the right is leaf roll 1, the one on the left is leaf roll 3. I think I could confidently walk down most vineyard rows now and put my hand on my heart and tell you that's leaf roll 1 and leaf roll 3. I don't know for the people in the room here and those in Margaret River if you could do that. Takes, you know, as I said, we've walked a lot of vines and it's just nice to get some photos now to show the differences. But really that leaf roll 3, see that really deep burgundy colour of the leaf is very, very typical of leaf roll 3. And, you know, you can those bright green veins, which you don't tend to see so much when it's talking about leaf roll 1. So we don't tend to see that curling of the leaves, a lot of that, you know, the name itself, leaf roll 3, sorry, leaf roll, you think of rolling. We don't tend to see that here in the limestone coast very much. It would have to be a very, very severe infection to see that. So for those of you who drive up and down the strip regularly in Coonawarra at the moment, where we've got a lot of young plantings across the district, we did see a lot of this reddening coming in very late in the season. So this type, you know, I'm talking end of April, early May. It was interesting because Kath and I were getting worried. We're going, why are we seeing this come in so late? Yes, it's getting cooler. Is it just a cold effect of those sorts of things? Anyway, just to let you know, when we sent those leaves off, that's coming back positive for leaf roll 9. Once again, this is Cabernet. So just be aware that, you know, that's what a leaf roll 9 vine will look like potentially. And then Vaughan and Fiona both put up around nutritional issues. Something we have noticed in this region is the issues around phosphorus deficiency, magnesium deficiency has definitely become more of an issue. I think that's a reflection of the type of industry we're in at the moment. The fertiliser tends to be the first thing that gets cut out of any budget that a farmer has to think about. Certainly I know, having just put phosphorus on the farm at home, how expensive it is this year. So it's certainly something to be concerned about. But what that does is it makes it really difficult when you walk out into a vineyard and you're trying to identify, is it a virus or is it actually just a nutritional deficiency? So things, this on the left here, this one here is actually been tested positive for leaf roll 1 and leaf roll 9. But then across, literally on the, you know, walking down the row, that one on your left was on, yeah, I'll say on the left, and then the one, that vine there on the right, same row. That's a nutritional deficiency that you're seeing here. So you can see that mottling on those leaves and there's a bit of the reddening. So when we did the test, we took leaf tests, we did green leaves versus red leaves, and we came back and that was actually very deficient in phosphorus and potassium. So you just need to be very conscious of that when you go through. I think when you look at a deficiency in a vineyard, you'll be thinking to see it pretty much throughout the whole vineyard. Virus will be quite, here's a vine, it might have its next door neighbour. It won't be as widespread as if it's a nutritional deficiency. So just to show you some of the complexities that we're dealing with in these regions. And then of course you get a vine like that, where you've got a nutritional deficiency as well as virus all in the one vine. I had to take a photo of this vine when I found it, but take note as well, there's a huge irrigation leak under that particular vine. And I think what's going on there, that's had so much water that it's leached out a lot of the nutrient component, as well as the fact I have started to notice where we have got a lot of irrigation leaks, get a lot more moisture around. We think that the vectors like that sort of environment, and potentially that's why we see a lot more virus expression where we see irrigation leaks, something we've really noticed this year. But some of the anomalies that we are also seeing, and I think we saw a lot of this particular symptomatic visuals this year. And, you know, initially we thought it might be GVA, and then we'd send a few samples away and they came back as not having any virus at all, not even the rupestris stem pitting and stuff. So, and it wasn't damaged, and yet those vines have been tagged. So, you know, we are hanging a hat on getting this right, and then you get things like that, but it doesn't look like your traditional leaf rolls either. So really would like to know what's going on in that vine. But then in these vines here, this came back positive for leaf roll 3, leaf roll 9 and GVA. And, you know, very end of season, still had, as you can appreciate, we were still picking fruit here into May. I think this photo was taken in that last week of April. But, you know, that's not showing your typical leaf roll symptoms that I showed from a few slides before, but it was also much later in the season. So we've really gone into that autumnal, I've had enough, I'm just shutting down type thing. But yet you can imagine that potentially looked like that vine a few weeks prior. So just those are sort of things that we're dealing with. Something that we did see a lot of this year, and I noticed, I think it was in Vaughan's slides, Kath and I, and that's Kath's fingers there, that we're driving through and seeing a lot of this. And, you know, you're not your typical green veins that we would expect to see, but that's that very Burgundian type redness that we're seeing in this leaf here. And what is this? And this was probably, actually, this is back in late March, and we were calling this like a liver spot. You know, when you get old, you get liver spots. That's what we were sort of talking about here, and you can see it in this vine here. And really, I think that photo went across to Vaughan. What in the hell do you think this is? Do you see a lot of this? He came back saying, oh, it looks like leaf roll 2 potentially, but anyway, it's come back positive for leaf roll 3. We think that that's just the evolution of the virus, as that will go to a more mottled effect. That's just in these younger leaves up here as the virus spreads from down the bottom up to the top. So once again, I don't know, did many people see that type of symptom this year in their vines? That, yeah, so just we hadn't really noticed it in previous years, but I just think being such a symptomatic year, maybe that's why we did see it. We've already had other speakers talking to these. That's a Shiraz vine here, obviously very late in the season. It's still holding on to its leaves. So we know that other red varieties can carry these viruses as well. And this is not one of my photos, but this is Malbec. And I reckon I've seen some Malbec that looks like this as well, further north from here. So I think just be conscious that it's not just Cabernet, although because I think we live in a Cabernet-based region, as you do over in Margaret River, other varieties will see the same issues. And finally, Shiraz decline. We have this in Coonawarra. I just want to alert this to the fact this is present in our region. You need to be aware of this. And this is a vineyard where these photos are taken. This is just early on the season. You can see here, very uneven shoot growth. And then this is later in the season. Those vines are not that old and never lignify. They never ripen fruit. And that's, you know, you get that classic curling of the leaf in this case here. So very much an issue. And this has come back positive for leaf roll 1, 3 and GVA. Cross it with a Shiraz and that's what you end up getting. So that's the sort of things that we deal with on a regular basis, and that poor vineyard's looking pretty average. So I think that's all from me for the virus identification. It's really just a short snapshot, local pictures to give you an idea. We are seeing this very predominantly across the Limestone Coast.
Thanks, Kerry. Great, good. I like that green room. I like what Hugh and Lara are doing there, helping us make it all smooth and seamless. Thank you. Monica Keogh is next, and she'll be coming in from Margaret River. I will introduce her, and then we'll cross to there. So Dr. Monica Keogh works for Department of Primary Industries and Research and Development, Western Australia. Dr. Monica Keogh is a plant virologist and molecular plant pathologist working for Western Australian Department, DPIRD, in the Diagnostic and Laboratory Services section. Monica and her team's current work focuses mainly on the development, validation and use of molecular methods for plant disease diagnostics across a broad range of broadacre and horticultural crops. Research interests include viruses of grapevines, vegetable crops, pulses, and oil seeds. She is also interested in supercomputing for plant disease, diagnostics, and the use of portable sequencing for rapid diagnostics in plant pathology. Great, over to you, Western Australia.
Thank you. Here we go. Hi, everyone. Thank you for the introduction, and thank you for having me here today to speak to you about some of the work that we've been doing here in Western Australia. So while I'm giving this talk, I've also got Tony Chinene here in the room with me, who's done a lot of the work with me on this. And yeah, so I'm going to talk a little bit about two previous projects looking at some diagnostic tools and one project that had a look specifically at some Chardonnay. So these grapevine leaf roll projects, I won't spend too long talking about the background on the viruses. I think that Fiona and Vaughan and Kerry have already done a pretty good job on that. But in WA, the focus has really been on the leaf roll 1 and 3 in recent years. And the symptoms that we already know about, the downward rolling of the leaves and reddening in the red varieties, you know, they can be pretty helpful as a diagnostic. And as Vaughan mentioned, you know, the white varieties can be more of a challenge. And that's why diagnostics can be really useful and really important. And it's also really important that we keep trying to improve when we can. And the other thing, of course, is that we don't want to forget about the vectors. And I think I might be experiencing the first technical difficulty of the day, that the slides aren't advancing. But yes, so the vectors are also important. Thank you. And also something that we should consider when we're looking at diagnostics. So in Australia, we've got the mealybugs and the scale to think about, and then grafting is also one of the, well, you know, one of the ways that these viruses can be transmitted. And also the symptomless infection that causes us so much grief. So knowing the damage that these viruses can cause and that mixed infections are also quite common really adds to the degree of difficulty. There was a survey published by Fiona and her team some time ago now. Out of 218 samples, 13% had leaf roll virus 1 and 14% had leaf roll virus 3. There's a couple of examples there of the infection. The first project that I was involved with was a, I was fortunate to receive a science award in 2018 to look at grapevine leaf roll viruses 1 and 3. Yeah. Thank you. And so the aim of that project was to have a look at virus diversity. So other than I think the survey that Fiona's team had published a little while ago, there wasn't an awful lot of published data on Australian leaf roll 1 and 3 in terms of the genomics and the sequences. When you looked on the public databases, there were no leaf roll virus 1 whole genomes for Australia. There was one for grapevine leaf roll virus 3. The New Zealanders have been doing a pretty good job. They've got lots of genomes up there. And so we really wanted to have a look and see, at least locally in Western Australia, what the situation was here and how it compared to elsewhere. We wanted to look at molecular methods for detection. So until a few years ago, if you sent your samples to DPIRD for diagnostics for grapevine viruses, we did all our testing by ELISA. We now do it by PCR. And that's as a result of some of this work, which is really good. And we also wanted to have a look at in-field detection methods, so the LAMP PCRs, which are portable devices. And this was all looking at just leaf roll 1 and leaf roll 3. And it then coincided with following on from that, we had another project looking at Chardonnay to look at wine. So in Western Australia, we produce some really nice Chardonnays, particularly from...
Just click another advantage.
Okay, don't do. Okay, sorry. And so particularly from the Gingin clone of Chardonnay, it's considered to be one of our sort of premium drops. And it's widely thought that the presence of grapevine leaf roll virus 1 actually contributes to the unique characteristics that make it so good, which I find as a virologist to be a really interesting problem and an interesting prospect, given that, as Fiona said, a virus infection is for life, and we generally consider that to be bad. So why do we want to look at the diversity and look at the genomes of the viruses? Well, I suppose a bit like COVID, this year we would have heard a whole lot about sequencing and genome sequencing, and when we have an outbreak, people want to have a look and see where it's come from, what it's related to, and they use it for tracking stuff. It's kind of the same principle, right? Where we want to be able to see what strains of the virus we have here, how they're different. We also want to make sure that our diagnostic tools are the best that they can be and that they actually pick up all of the strains. So one of the reasons we had to move away from ELISA was that I think Fiona mentioned a lot of the anti-sera wasn't actually picking up, necessarily guaranteed to pick up all of the possible strains that could exist. And we hadn't really looked at the genomes to see what we had here. Having access to whole genome sequences means that we can design those tests to be really, really specific, or to be broad if we want, make sure that they pick up all the strains, or if there's a strain of concern, we could just pick up one if that's what you wanted. And having the whole genome gives us the best chance of success to design something that's really, really good. And the really nice thing from my point of view is that the viruses are really small compared to, say, a human genome or something like that. It's, I say, relatively easy to kind of put them together and to get those answers. So from this work, 264 samples from 11 sites, we tested them by PCR. We tried to get a range of varieties where we could. I should point out that these numbers might look scary, but it is biased. I did walk onto people's properties and say, show me your worst. I was actively looking for the sick stuff. So it was really good that we were able to get hold of some leaf roll 1 and 3. And out of this work, we got 12 new whole genomes for leaf roll 1 and 13 for leaf roll 3. The leaf roll virus 1, I think they'll probably be the first ones from Australia, but I know that Fiona's team's also been sequencing grapevines. So in the very near future, hopefully, at least I hope to get these ones out in the public databases soon. So what we did with this was that we went to look at some in-field diagnostics. So we're doing PCR in the lab now. And yeah, one of our comments is, time. So, it takes time to take samples, send them, we receive them, they get tested, and to get an answer. And, we know that when people are walking their blocks and having a look at things, you want to know what's there. So one of the possibilities is to explore these in-field detection methods. So we used those genomes to design a test for leaf roll 1 and for leaf roll 3. As far as I'm aware, based on all the sequences that are available in the public databases, this test that we've designed should pick up everything that we know that we have here in Western Australia. But that's not to say that we've definitely covered off the true diversity. If I had the chance, I'd like to do more sequencing. This test works on crude extraction material in the field. So instead of having to take it to a lab and spend a day extracting the RNA from it, we can put it in a tube with some buffer, some leaf material, and some steel ball bearings and shake it, and then use that to put into the device, which is here. It's just one of the types of commercially available devices you can buy that do this sort of thing. And it will give you, from when you start the machine to when the run finishes is about half an hour. And so it can run off its own battery, which means that I could set it up in the field, literally in the back of the car. as an example there. So with a couple of these machines and a fridge for keep the stuff cold, I was able to get through probably a couple of hundred samples there, but that was probably pushing the capabilities of it for one person in a day. I think really the true value of something like this is, you know, the one-offs, the things that you're not sure, the things that look a bit weird, you know, maybe like what Kerry was showing, the really deep red symptoms at the end there where they thought maybe, maybe not, you know, that sort of thing. If you've got hundreds and thousands of samples that you want to test, this probably isn't the way to go. You know, probably having access to a lab, any one of the labs that Fiona showed have some sort of high throughput capabilities, and it's probably better to talk to them about a deal. An example of the result that you get, literally this is the output from the machine. So you see a time to positive, which tells you that you've detected something. And these temperatures are also important. They need to be within a degree of what you expect. And you get a graph. So if it's not infected, then it's just a flat line. The ones that are infected will give you this nice curve. And they also show you the peaks for the temperature. So this run had both leaf roll 1 and leaf roll 3, which is why the two peaks are different here. And so that brings me then to talking about one of the ways in which we use the tool. So following on from that project, we managed to get a regional incubator project to look at Chardonnay, specifically to look at the Gin Chardonnay. As I mentioned, it's one of WA's sort of premium kind of wines, and the presence of leaf roll 1 is thought to contribute to you know, why it's so good and so special. But it does, I'm sure it does also contribute to the fact that, you know, typically the bunches of Gin Chardonnay, you know, they're uneven, they're smaller, there's reduced yield. And we really wanted to know if you could tell the difference between wine made with leaf roll 1 and without it. The one snag was that we couldn't find Gin Chardonnay that didn't have leaf roll virus 1. So in the end, we did the work with leaf roll virus 3. So vines infected, vines with leaf roll 3, vines without. And one block was Gin Chardonnay and the other one was a clone 95. So it was pretty interesting to start with. We went out, and I should say we had some really great support from some local growers down here in Margaret River and from Richard, who made the wine for us. And we, first of all, went out, we collected a few hundred samples across a couple of blocks, basically individual vines. We tested those by PCR back in the lab, narrowed it down to about 30 or 40 from each place that we thought we would follow throughout the season. And then we visited a couple of times and just checked using the lamp on site, checked that those vines were still, well, the ones that were positive were still positive, and just checked that the negative ones were really negative, or that we couldn't detect the virus in those ones. And then we came back, they're pretty good. They let us in sort of a day or two before the big harvest happened, and we were able to get the fruit. Richard did the small batch winemaking, and it was really interesting. We only had a few people help us out with the sensory evaluation. Statistically, it didn't show any difference, but there was one person who was able to pick it almost all of the time. So for a pretty small study, I know we'd be pretty keen to do some more. One of the most common questions I got was whether we would be extending it beyond Chardonnay. And I'm pretty sure there's interest to see it across some other varieties as well. So from that, which leads me on to the next bit, which is new diagnostic tools. And I think one of the most exciting things we have going on in diagnostics at the moment is the use of high throughput sequencing. It's definitely been more in the public domain and with COVID in the last year, which has been pretty terrible, but I think we've been pretty lucky in Australia with what we've had to deal with. As a virologist, I've had to contain myself at times because it's been super interesting for lots of different reasons. But I think the use of tools like sequencing has really come to the front, and I think, yeah, being able to use them in plant pathology and in virology has been really exciting. So what you see here is called a MinION sequencing device. It is really that small and literally fits, you can hang on to it. It's USB, it's a USB device, plugs into your computer, and it runs sequences. So before this came along, if you wanted to do high throughput sequencing, you'd need access to really quite a large device, someone to run it. It's pretty expensive. They can cost hundreds of thousands of dollars, if not more. And most labs don't really have access to that. So this thing is much more affordable, and scientists can have their own. It's also been used quite high profile, so in Ebola, Zika virus, and now COVID, for sort of real-time diagnostics, real-time determination of strains. It's also being used in agriculture. So Tony and myself were involved in a project in East Africa, the Cassava Virus Action Project, where we were using this device to determine the correct strain of virus that was present in a location so that growers could be provided with clean planting material with resistance to the correct strains, which is really important in increasing yields for them. So there's lots of different ways that we can be using this. I won't spend too long on this, but the main point is that this part in the middle here is the disposable flow cells. So you buy those, they get cheaper as you buy them in bulk. So the device itself is about $1,000. Those flow cells, different types of them, range from 100 up to 1,000, depending on how many samples you want to put on them. But we thought, if people are using this in a medical setting, they're using it in agriculture already elsewhere, we should be looking to use it here. It's really quite powerful. And part of this project that Tony's working on is we're looking at a way to redesign the way that we're testing for the grapevine viruses. So at the moment, if you want to test for, I think, probably 13 or 14 viruses, or the full panel of viruses that Fiona mentioned in her talk, that takes a lot of time. You're going to run a PCR, and in some cases 2 or 3 PCRs for every one of those viruses, right? That adds up, takes time, and it costs money. So our goal was that we wanted to see if we could do it in one test. So we're still working on it, but we think it's really promising. And one of the things is that data analysis really comes into this. So these little devices are producing huge amounts of data, and that's probably where the bottleneck is. But it's not just viruses, but IDing the vectors as well. And the other thing I didn't mention was that you can take this in the field. So, I mean, it's a bit fiddly, but you can take it to the place where you want to do the ID, whether it's in the field, the hotel room at night, whatever it is that you need to do. And also gives us the option of looking maybe even at the viruses in the vectors, which is another aspect of it. So, like I said, there's lots of different ways to use it. We can use it in the lab, in a laboratory setting. The stuff we take out in the field is slightly different. In a laboratory, we'll use kits like this. It might take us a day to extract the RNA, a little bit less if it's DNA from the vectors, RNA for the viruses. But we've also picked up this other machine, which is, I probably should have bought that for, it's quite small as well, but it can be battery operated, and it will give us DNA in 30 minutes that we can then use on the sequencer. So that's also a really big improvement in recent years. So unlike the LAMP machine, where we can do a crude extraction, just shake up a leaf in some buffer in a tube, that's sort of a little bit too crude for the sequencer. So we do actually need to do a bit of a process to get some clean DNA and RNA out of that. But the time savings are really good. So Tony's done some really nice experiments on some mealybugs. He used that PDQ-EX device, that device that we mentioned, so 30 minutes or even less, I think 20 to 30 minutes to get the DNA. He spends 15 minutes doing some preparation steps, and then that goes onto the sequencer. And then probably within an hour like that, you can get the sequencing as it comes off the device. So it's in real time. You can be looking at it then, or you can wait for the run to finish. But if you really want to know what's in there, you can look at it straight away. And so it's shown here that he can successfully identify these these bugs. So this project also looks at some other targets. So there's some medfly and whitefly in there as well as the mealybugs. This is one of the things that we've struggled to get some samples for. So if anyone, seems everyone's got really clean vineyards, we've struggled to find some mealybugs, but if you have any issues, give us a call. We'll be happy to come and take some of them away for you. But I think it's really promising. If we were to do this in the laboratory by standard methods, it'd probably take at least a week. We would have to send away the PCR product to be sequenced by a third party. So we can really do this in day and a half kind of turnaround. So I think that's pretty exciting. When we look at the viruses, it does get a little bit more complicated. If we have a sample of grapevine, it's a bit weird. We're not sure what it is. It's not coming up for the standard tests that we do. We can use this device to sort of have a look. The whole genome sequencing approach, where we sort of sequence the whole lot, it does produce a lot of data. So we have some pretty big computers that we can use to crunch that data quickly, but they're not conducive to the field as such. But at least we can get the device producing the data so that it's ready. And that 15-minute kit that I referred to for the mealybugs, that's also not really appropriate for this at this stage, because working with the RNA is a little more difficult and it takes longer. So in a laboratory, to do this kind of process would take 2 days for the sequencing. But we have come up with a bit of a plan for an alternative test. And this is what I said, we wanted to redesign how we test for grapevine viruses, is that if we run a PCR for all of the viruses at once, so Tony's done some work on being able to run the PCRs all at the same temperature. He took all of the genomes that we could find in the public databases. So we tried to make sure that what we were designing would pick up everything. We wanted to make sure that we weren't going to miss a strain or something because a sequence was a little bit different at that spot. So we've tried to be as good as we can be based on the information that we have. But like Fiona said, these things are much easier to manipulate than an ELISA. So if next year someone publishes a whole bunch of genomes and they're really different and it shows that our test isn't going to pick that up, we can actually change it relatively easily, which is really good. So at the moment, well, a few years ago at DPIRD, if you sent your samples to us, like I said, it was ELISA and it was only Lifrol 1, 3 and A. Now we're doing PCR as well as 1, 3 and A. We can do 2, we can do GVA, we can do GVB. There's a few others as well. So we're not quite to the full suite yet, but give us another year or two and I think we'll be there. And that's for standard PCR, but I really hope Tony has about 10 viruses working with this new method. So he'll do 1 PCR for the 10 viruses. We put it onto the sequencer. So instead of just getting a yes, no, we get a piece of sequence. So that's really cool. And actually the biggest challenge is, like I said, it's going to be the data. So one of the good things that's come out of COVID is that so many people are using these devices. There are lots of people also doing really cool things with software. And so being able to analyze it in a way that's reproducible and that it's repeatable and that it's accurate is going to be really important. But so I hope that with this tool, we will both be able to use it in the laboratory to provide results for more viruses at the same or cheaper price than what people are doing now. So the different types of consumable flow cells are really helping us with that. There's new ones coming out all the time. The small ones, they're about $100. Like if you could run 1 sample on that tested for, 10 to 15 viruses at once, you're going to be ahead of the game. And so that's what we're thinking. That's where we're going. It is a work in progress. But in the meantime, we're still offering our PCR testing. So one of the things that has been a challenge, not being able to get out as much last year, has been a challenge to obtain known positive samples for validation. So now we can move around a bit more. Same as for the mealybugs. If anyone's got something weird they want us to take a look at and they're happy for us to use it in validating our protocol, then give us a call. COVID-19 did slow us down a little bit. COVID is an RNA virus, and so a lot of these grapevine viruses. So unsurprisingly, delivery of these reagents and kits that we use are the same they use in medical labs were prioritized to those other labs. So we can't really argue with that. And the field extraction for RNA is definitely the one that's giving us a little bit of grief. That device that we used for the DNA doesn't quite have a kit that's good for the plant RNA just yet, but we're exploring some other options, and we're pretty sure that we can get that happening soon. And so, as I alluded to earlier, so the small device that's USB, that handheld, needs to be plugged into a computer. And just recently we've come up with this new one, which is the same thing. So it's got that device in the bottom of it, but it actually has a screen, and it actually has the computing power in it to do a good proportion of the analysis, which is really exciting. So that's the bit that lets us take it in the field. That's the bit that's going to let us take it out and do it in the back of the car like the other stuff. So that's where we're at the moment. And yeah, look forward to some questions later on. This is just another picture of someone using it in the field. And like I mentioned, this project that we're working on also looks at viruses and vectors in some other crops as well. So this isn't just useful for grapevines. It's hopefully going to be applicable to lots of industries. And over the last few years, we've had a lot of help from a lot of different people. So I thank the team at DPIRD and to the people who've led us onto their properties and to make wine from their crops. And some of the DDLS clients who've been really supportive in letting us use their samples to validate across different methods. Thank you.
Thank you, Monica. Great. Next, I'll introduce Jen Venus. Actually, she'll introduce herself because I don't have a bio for you. So if you come, and while she's doing that, I'll set up your presentation. So
lovely. So I apologise for not sending through a bio. I've recently changed jobs, so it's been a little bit hectic over the last month. So I now work for Accolade Wines in the Adelaide Hills, covering Clare and Tasmania. Previous to that, I was with Brad Case Contracting in Langhorne Creek, looking after quite a wide region there through Langhorne Creek. And prior to that, I've been with Nutrien and Fable and various other things. My passion, I suppose, outside of all those jobs has been trying to get a solution for scale and mealybug. I am not an entomologist. I'm an interested viticulturalist that saw a problem and we still haven't fixed it. So the great thing about being so far through the program is that most of what I'm going to talk about has been covered to some degree. So yeah, so been in the industry for probably 20 odd years. Yeah, so it's exciting times, and it's great to see that Monica's doing some stuff we can take into the paddock, because that's what we need to be able to do. So I think we're nearly up to the speech, the talk. Rightio.
this is your mouse.
My mouse, thank you, my timer. Rightio. So I was given the opportunity to speak about vectors, vector. What are key vectors and when are they at highest risk in the vineyards? So really, let me just see how this one, I'll just go down.
Just use this mouse. Yep.
So just go down.
And look over there. Yep.
Where do I hit it down? There.
Yeah.
So really, I thought I'd start by setting the scene, because I thought we would get a lot of science around this. So I thought I'd go back really to a grower's perspective around virus. And what does a virus mean to a grower? You know, locally, probably industry are looking for lower yields in premium regions. And as Monica said, gin clone of Chardonnay full of virus. Is that a bad thing? We don't know. From a science point of view, yes. From a grower point of view, you know, we still are trying to struggle with that, I think. Impact on ripening. Are growers noticing that their Shiraz or their varieties are ripening differently? Is that a problem for the winery? Vine health decline. Our vines are pretty old now and they are full of viruses, a lot of those older ones. Is that a problem for growers? And, you know, I think it became a problem for growers when we went for a rapid change of varieties. When we went from, say, me, Langhorne Creek, from a very big Chardonnay growing region to a Shiraz growing region, we top worked a lot and GVA all of a sudden was at our doorstep, and that was a problem because all of a sudden we didn't know we had to virus test, or the risk around those viruses wasn't known, and when we top worked, it was a disaster. So all of a sudden growers now are a little bit more aware that viruses might be a problem for them. You know, when we come out of a research background, they're a problem. If we're in a big corporate organisation, we can see that and manage it. For everyday mum and dad grower, viruses are really hard and they're probably fairly low on their risk assessment. So, you know, we need to think about that when we're delivering this information to growers. Worldwide, so I did a bit of a search internationally, you know, as it says in the top line, as a wine grape grower, we have a lot of challenges in this day and age, and the effect of viruses are becoming of greater importance as we start changing things over, looking to where we go for the next 25 years. You know, in Canada, it's a $9 billion industry, 23 million bucks is lost annually to viruses. Do we know what our loss is in Australia? In the US, different viruses, fruit's being rejected. You know, the wineries won't take it because of uneven ripeness. And I think the other thing is that the solutions are fairly simple. We rogue it out. But really, that's probably suboptimal. The growers don't know whether it's got virus or not. We've had some really great experts here today going, I think that's a virus, but maybe it's not. Once we get Monica's stuff in the paddock, that will change the game for us. So that's sort of where we're at the moment. So I'll get back on, I've had my rant now, so I'll go back on to what I was told to talk about. What is a vector? So a vector is something that transmits a plant virus. Generally, they're insects. So with the mouth part, as has been talked about earlier, aphids are probably the most common in tomatoes, spuds, all those sort of crops, not a big problem in grapes. Leaf hoppers, as we've heard from internationally, a problem. For Australia, every talk has spoken about mealybug, scale. Nematodes a little bit can be, and obviously now mites when we talk about Pinot Gris virus. So they're our big keys. Unfortunately, that's covering, it's a fairly basic, like a mosquito in a human. An insect sucks one vine, it moves along, it feeds from another vine and it transmits that virus. So it's a fairly basic sort of mechanism. Some insects ingest them and then moves it that way. A lot, the virus stays on its mouth parts. You know, it moves within 48 hours and reinfects the next vine. So I think we've covered that a fair bit already in what we've spoken about. Symptoms have been gone through pretty well so far, in that, you know, there's a lot of reddening in our vineyards. There's a lot of distortion, curling, they're sort of viruses, but there's lots of other things that cause that. So we really need to train people well, as we've heard previously from Kerry around, you know, they've walked a lot of Ks to work out what a virus is. So we really need to go back and train people around how to identify viruses in the vineyards. So, you know, I just thought I'd bring, have I missed a slide? Don't know if I can go back. Can I go back? We'll go with this one anyway. So this, I just thought around that, what is a virus and how do we know? This was the Adelaide Hills last year. Obviously, we had fires through the hills and our weather and environment was quite different last year. This was a patch that turned red after harvest, and by the end of sort of autumn, the entire vineyard was red. We had people on the side of the roads painting it. It was so stunning. And this year, there is none of it. So, you know, this is probably an environmental impact from last year with the changed environment, the fires, the heat, that this is probably not a virus. We have done a little bit of work around this, but, you know, there's a lot out there that we don't know about, and trying to identify it is really difficult. So getting on to what is virus, probably, as I said, for Langhorne Creek, the first problem was GVA. This is one of the first blocks that was grafted in Langhorne Creek from Chardonnay to Shiraz. It took 4 years for that grower to work out he had a problem. and then the whole block was removed. That is fairly costly for a grower to get to that point. So this is even before we talk about vectors. This is just, you know, transmitting it through graft union. So we really need to get to growers to say that this is a problem and that they need to do more work, and we need to get it cheaper for them to test. So now we get on to the proper one. Sorry, there's the eyes wrong. Vectors. So what are the vectors? What are the problems we have in the vineyard with vectors, and how do we solve that problem? So obviously for us, it is scale and mealybug are the main vectors that we have, with mites coming in there with the Pinot Gris virus. What do these things do to our vineyards? So this is what growers see. This is what growers see. This is a consequence maybe of the vector or of a bad grafting, but this is fairly low on the grower's outlook compared to this. Crop loss from scale and mealybug is immense. This is, you know, mealybug in a bunch. You're not going to pick that. So this is what growers see, and this is where we started, where I started targeting it at growers, and now we've really got on to the virus side of scale and mealybug, that they are a major problem for our industry, and we are doing very little to solve what is a major problem for our industry. That these are two very problematic vectors for virus, and problematic for growers in terms of the impact they have on the quality of their fruit and their vines long term. We've seen this a little bit, I think, of what the mealybugs are. So this is the long-tail mealybug on the back of a leaf. You know, if you squash them, they're a yellowy colour. They're quite visible and easy to see for monitoring. The obscure mealybug is shorter. When you squash that, it's more yellow. But once again, quite easy for a grower to see. On the backs of leaves, we tend to see them after flowering. The reason I say that is that all of our products to control mealybug and scale in Australia have to be used before flowering. In New Zealand, they've got better options for products that can be used post-flowering. In Australia, we have nothing. So we are fighting a battle against vectors, insects, that we really have nothing to control them with. The products we do have, we have to use at the wrong time. So I'm really trying to change that for the industry, but it is a slow process to get products suitable. Insecticides are a bad word in any industry. No one likes insecticides, but we really need to think strategically around how we control these problems. So that's mealybug. They do produce a honeydew similar to scale, but nowhere near as much as you'll see in the next few photos. So when they're feeding here, this is probably not the worst phase of a mealybug. We'll see in the next one. Here you can see all these yellow little dots. They're the crawlers hatching out, or they're actually born live. That is the problem for mealybug, is we get 6 generations in a year. When they're this age, they move very quickly through a vineyard, on the wind, running, moving through people, through spray carts. At this phase, when they're when they're crawlers, very mobile through the vineyards. So very easy to spread virus through the vineyards. If we look, I've just done it on long tail here, but pretty similar for, I better not move away too much from the camera, very similar for obscure. You know, the female produces around 200 live young over a period of about 2 to 3 weeks every time they go through a life cycle. The life cycle is about 6 weeks, very dependent on weather. So we can get a fair few of those in a season. So you can imagine we can use some products, chemicals early before flowering. If you miss that, you've got a lot of generations of mealybug that occur after flowering that we can do nothing about. So you have to be fairly rigorous with your monitoring early to reduce the number of mealybug or post-season. So, you know, 3 to 4 generations a year, that may go up depending on the temperature and what kills them. If we go into scale, I've put this up because this is October 12. I don't know whether you can clearly see all the scale that are on that vine. This was probably 11 was the first year we realised we had a problem with scale in the wine industry. 2012, we started monitoring. By that point, there was a major problem in the industry. So these are the females that are mature in a vine. This is the impact on a Sauvignon Blanc block. Nothing related to virus, I know, but I think it gives us an indication of what these vectors are doing for our vineyards. We are at odds as to whether this is a problem for winemaking, but I don't know many winemakers that would want that coming into their winery, or that. I'll go to the next one because it's better. They produce an abundance of honeydew, which then just drips off your vines, and the sooty mould comes in. But you can see they are abundant feeders scale. They really start from the start of the season and they feed incredibly throughout the season. And so the ability for them to spread virus is very high. And the other thing here you can see is this bit at the back was treated for scale. This bit wasn't. So there's certainly a delay in bud burst where you've got high numbers of scale as well. In terms of when there are a risk for you as for virus in particular, a scale, this is laying eggs, they generally hatch right on flowering. So we can monitor and then generally pretty close to flowering they hatch. A problem once again for us because we've gone past our insecticide withholding periods at this point. So we're trying to manage these earlier with products that aren't ideal at that point. I did have a lovely little video in there, but I was worried about the Western Australian link. Between that hatching and getting to this sort of first instar, they're a crawler, and you can literally see them running up and down wires in your vineyard. If you watch them when they hatch, they're extremely mobile. They come out and they will move with the wind. They will run literally up and down the wires. So at that point, they feed a lot, they're moving a lot until they settle onto the back of leaves. So a really high risk there for virus. Once they settle on the backs of leaves, they do tend to stay where they are. So probably once we get through that first burst in November. But, you know, Fiona, I think, spoke about they have one generation in a season. Scale have far more than one generation in a season. I think you've probably all seen scale hatching in December, in January, at Verazon, through. So not all of those will live through, but they're all creating crawlers that are feeding. So majority of that hatch in October, but we really do see hatchings all through the growing season. So that is a problem for most of us. So if we go on a bit, this is a really big problem for our industry, is that we're doing a fair bit of top working, grafting, changing varieties, and this, you know, what we're grafting onto there has a lot of scale already on it. The options for the growers to control that is pretty limited. So we're converting over, putting on beautiful new budwood onto stuff that's got scale or vectors already there. And I couldn't find my photo, a missing one, where we went through last year and opened up guards of a new planting to, you know, train them up, and there was scale already on those new plantings. So, you know, it is right from when we first plant those through to mature vineyards that we're getting scale coming in on those vines. We need to find a solution for that. There are two, just as a bit of an education, there is vine scale and there's frosted scale. Does it matter which one you have? Probably not. They both are a problem in vineyards. They'll both spread vectors. The problem for us, as I said, you can see here they've walked around and they're back on the mature female. This phase here is the vector problem predominantly when they hatch. Here, they're very active at this first crawler stage. Once we get down here, they tend to settle on the leaves. This is through winter. They overwinter on the spurs. This phase here of growing to maturing and laying eggs takes about 6 weeks. So this phase here is what is the biggest concern for viruses. I'll skip through those couple. And you can't see, unfortunately, over there. But the other thing, you know, mealybug lay or have about 200 young per female. A mature grapevine scale will lay about 1,000 eggs per one of those little shells that you saw in those first photos. So if you've got a small population of scale in your vineyard, it can explode very rapidly and spread very rapidly if the conditions are good. Frosted scale is about 400 eggs per mature female. So there is a great opportunity for that small infection to spread quickly if we get the right weather conditions. We talked about chemicals. Interestingly, New Zealand has just banned these, the group 1s, the chlorpyrifosses. I assume that means they'll ban tocothionin and supracide and maldacin and all of that group. They use this one a fair bit. Movento was the one that Vaughan spoke about. For us, that's an EL cutoff of 18. In New Zealand, it's 26. So they've got an opportunity to use that later when it actually is better positioned than what it is for us. This one here, Applaud, was the other one Vaughan spoke about. We have a cutoff of 80% capfall. In New Zealand, it's bunch closure. So I think it's EL 29. So more opportunity to use these strategically in a better position. We really struggle to get these chemicals positioned well for the insect life cycle. So what I mean about that is if we look at a Movento works on those young scale, those crawlers. So if those scale haven't hatched when we're spraying it at EL 18 and they're hatching 2 or 3 weeks later, we're going to control a very small population because the product won't work on those mature females. It needs to have crawlers. So we're really struggling with finding products that we can use at the right time. We did a huge amount of work on this one. In New Zealand, they have Confidor that they can use through winter. Really, we can't use Confidor in Australia. Clothianidin, which is the Confidor's little sister, probably. We did a tremendous amount of work on that, and really the industry is not overly keen to make that part of our management system. So we are still trying to work on if you're replanting a vineyard, whether you treat it before you remove to try and treat those roots and kill mealybug and scale before you remove the vineyard, and then maybe when they're young. But really, you know, we can't use that. Trevor was a new one in. Still, it's a group 4, which are unfavourable in Australia. So we're getting some reasonable results with that, but not great. And obviously up here, these, you know, chlorpyrifos are pretty frowned upon. I'm about to ring them. Yep. So I'm nearly at the end. This one. This is the way we would like to go, as Vaughan said, with beneficials. Metaphicus is really great for scale. The Cryptolemus is the mealybug destroyer. It is really good for mealybug. It's not so great for scale. This is the way we want to go. Very expensive at the moment, and we still need to do a lot of research on it. Monitoring, I think most of you know now what to look for, how to see them. It's pretty obvious when you see all that sooty mould that you've got something in your vineyard. We'll just flick through. Usually they work side by side, so if you've got mealybug, there is often scale there. As I said, I do this as a sort of a interest for me, but we are now looking at why we have had an explosion of mealybug and scale in our vineyards. So sort of where we're at as an industry is reflecting on the change in our spray programs, the increased use of sulphur, and what we have changed to cause this problem in our vineyards. So I've had a little break from doing scale work for the last couple of years. I'm really keen to actually start again and get some interest and get some overview of why we have the problem and what we can change, how we can get beneficials back in, looking at what Vaughan's doing to really not rely on insecticides. That is not the way forward. We need to review what we're doing and we need to get rid of these vectors out of our vineyards. So I think the last little bit, this one, quick. Blister mite, those sort of things, most growers would walk past that and not care. Most growers go, they're there, it's not going to be a problem, because it hasn't been a problem for a long time. So once again, that's going to be interesting for the industry of controlling those. Obviously, the spread of virus through vectors, mealybugs, is a major concern for the Australian industry. And I think the thing we've got to remember is that we need to start clean and stay clean. What you put in the ground when you start is there for 30 years. If it comes in dirty, it's dirty for the rest of the time it's there. And if you already have vectors in your vineyard, it'll be dirty pretty quickly. So start clean, stay clean. If you've got vectors in your vineyard, do something about it. Once you've got it, there is no really, no cure once you're infected other than ripping them out. That's expensive. So really think about that when you're establishing vineyards. That is it.
Thank you, Jen.
We'll go off. We're going to go to lunch now.
Oh, no, we've got another
Another one.
Another session.
Oh,
I'm gonna-
thinking we're up.
great. I'm going to ask Hans to come up and introduce the next couple of speakers as it's a real interest of his. I'm aware of that, and he can introduce himself as well while I set up Mickey's thing. So, Hans.
Thank you, Ollie. Good afternoon, Coonawarra, and good morning in Margaret River. I'm viticulturalist with Penley Estate, and for those who know me will know of my keen interest in precision viticulture. I'm also the fortunate recipient of a 2021 Wine Australia funded Nuffield Scholarship investigating practical data management options for viticulturalists. But where will the data be coming from? Monica has provided an example in her presentation, and I noted her comment about the volume of data she and her team are producing. And for the final part of this session entitled Vector Victor, New Technology for Vector and Vineyard Monitoring, we will add to this by directly asking, how can technology address some or all of the key virus considerations of healthy planting material, improving diagnostics, farm gate hygiene, vectors, and improving surveillance. As such, we've invited Drs. Mickey Wang from the University of Adelaide and Nancy Shellhorn, CEO of Rapid Aim, to provide insights to their research and technology. As an introduction, Mickey Wang has areas of research in grapevine virus, remote sensing and hyperspectral imagery, publications covering virus pathogens in Australian vineyards, and the accuracy and utility of low-cost thermal camera and smartphone-based systems to address grapevine water status. And as we'll see, the use of hyperspectral sensors mounted to drone platforms to provide mapped insights on the presence and distribution of virus in vineyards. Dr. Nancy Shellhorn is co-founder and CEO of Rapid Aim Digital Pest Surveillance and Management Systems for Agriculture. Rapid Aim takes the guesswork out of pest management. Prior to co-founding Rapid Aim, Nancy was a principal research scientist with CSIRO in Brisbane, where she developed and led concepts to achieve pest suppressive landscapes, a way of measuring, designing and managing agricultural landscape mosaics, for productivity and biodiversity. Nancy has served on many national and international committees and advisory panels, including the Federal Office of Gene Technology Regulator Technical Advisory Committee, the GRDC National Grains Pest Advisory Committee, the Cotton Industry Technical Advisory Committee, and the OECD Integrated Pest Management Advisory Committee. Dr. Shellhorn received a Bachelor in Agriculture from the University of Missouri, Columbia, a Master's in Ecology from University of Missouri, St. Louis, and PhD in Entomology from University of Minnesota in 1999. She joined CSIRO in Australia. Today, she is joining us to provide an overview. Today she's joining us to provide an overview of RapidAim's Internet of Things sensor technology, which is aimed at taking the guesswork out of pest management. Combined with Mickey's presentation, these technologies could cover two aspects of virus management: identifying where virus exists and monitoring for vectors. Fiona Constable noted infected vines are a source of infection for other vines, blocks and vineyards. And then with Vaughan emphasising the vine virus vector interaction. In terms of covering the area, walking the kilometres, can tech provide solutions to facilitate this in a vineyard manager's busy every day? So handing over to Mickey.
Hello, everyone. My name is Mickey Wong, and I'm the PhD student of Dr. Vinay Pagay from Adelaide University. Today, I'm going to talk about the PhD project I'm doing, which is looking at using remote sensing and machine learning technology for the grapevine viral disease detection. So the object of this project is basically I'm going to compare different sensors to look at this virus. For example, in here, yeah, it's been blocked. But anyway, I have this radio spectrometer, and I have this just normal RGB camera, or also the infrared cameras, and I also get a hyperspectral camera system. So what I'm trying to do is mount those systems on the drone, and then trying to do a large-scale scan for the vineyards, and then trying to use machine learning techniques to get reliable results. So as Dr. Fiona was talking about before, the virus is really difficult, it's very complicated. So we actually also treat different virus and different varieties differently. For in this case study, we look at the Shiraz disease, which caused by GVA. And then I have this video here, which is set a field camera in the vineyard. So basically taking photo every hour. And then you can look at the right-hand side, which is a Shiraz disease infected by GVA, and the left hand is a healthy vine. And you can see that in the early of the season, it actually had delayed bud burst and also delayed spring growth. But what's happening is after, in the middle of the season, those GVA infected vine actually, they could catch up. So about start from November, you will almost can't tell the difference between these two vines. As you can see here, start from November. So by using this unique character, what I can do is basically flying a drone over the vineyard in the early of the season in October. And you can see that some of the canopy are much smaller than the healthy vine. So I can identify them as GVA infected. And they are also other things, for example, trunk disease or dead vines or something. So we can also discriminate by taking another photo, which is in the middle of the season, for example, in December. And you can see those vines actually are catching up. So they are similar size as the healthy vine, but the trunk disease or dead arms, they never grow, they leave a gap there. So then I can use, then I can quickly just calculate the canopy size of each vine, and then you can quickly locate all those GVA infected vines in this block. And then I can provide a quick map for the growers to identify, to tell them where all the disease are, and also maybe the trunk disease somewhere like that. So this is just a simple application for a very obvious symptom disease. So someone maybe ask, what about a leaf roll 3 in Chardonnay? Because sometimes they don't really show in symptoms or no obvious symptoms. So in this case, I'm actually using a hyperspectral camera for this study. So to understand what is hyperspectral camera, so basically we need to understand the light. Light is electromagnetic waves. So they're all waves. So different wavelengths are different. So for example, in X-ray or UV light, they have much shorter wavelengths, but they have higher energy. And in the infrared, they have a higher, longer wavelengths, but less energy. But for human eyes, we can only see a very narrow range of the spectrum, which is about from 400 nanometers to 700 nanometers. So within this range, our eyes actually only sensitive to 3 bands, 3 peaks here. So which is, to our eyes, which is red, green, and blue. So people using this one actually developed the digital camera. So the most common camera, same as the camera at the back of your phone, they are all RGB cameras because they only need to capture those green, blue, and the red, green, and blue band, 3 band, and then can combination, or they can mix to make all other colors we can see. So that is enough for us to just take a normal picture for human eyes to see that. But you can see the problem here is actually, if you only select, you can only capture those 3 bands, you actually lose a lot of information here. So what hyperspectral camera do is instead of just taking 3 band here, it takes hundreds of band here. So you get a continuous spectrum of this range. So usually they can go from about 280 nanometers, which is UV light, to up to 2,500, which is infrared light. So it's far beyond human vision. So it can see what you cannot see. And also it can see in detail. So for example, you look at the GVA or leaf roll infect leaf in Chardonnay, they both look green to you, but you can't tell the difference. But hyperspectral is possibly in some of the spectrum range, they have big difference. So they can tell you there's a difference in this image. But the problem of this thing is because each pixel contains hundreds of numbers here instead of just 3, the size of the hyperspectral image is huge. So for example, if I scan about 5 hectares of land, it can produce about 50 or 60 gigabytes of data for me. So this makes the data analysis become so difficult. So, but thanks to the development of technology in computer, so we have now get a lot of machine learning methods for the big data analysis. For example, in here, we got a hyperspectral image here, and then we got different ranges, regions, and I can tell the computer says, okay, region 1 is leaf roll 1, region 3 is GVA, and region 1 is a healthy vine, and then tell the computer, feed information to the computer, and then computer should be able to analyze all the data and predict the rest of the pixels in this image. So then we can start to doing this spectral analysis. And more recently, for example, the neural network is even more advanced technology for the data for the image analysis. So this one has been used everywhere in the world. For example, facial recognition or all other things is using this technology. But this thing is really belongs to computer science. So it's really different world. It's too hard explaining here, but well, give a little bit idea about what this one can do is basically I'm flying a vignette and I just select the corner here and I put a grid line there so I know which one is which. And then I feed the information to the computer and the computer will do the classification. So it will classify those area, give the green color pixels. So okay, those ones are healthy pixels. And then the red ones are disease pixels. Then you can see that some area got more red, some area got more green. So then with that, I can provide a simple simple map to the growers. So just showing the row number, vine number, and then you get a map of your block about the disease status. Or I can even give you a score, like what's the percentage of this vine got a disease? For example, 100% of pixels showing this as disease, or only 50% of pixels showing this disease. So once the grower has this map in their hand, so they can basically have a better idea about the virus status in their block, so they can make a better decision in the future. That's all for me. Well, thank you, everyone. Yeah.
Thank you, Nikki. And yes, so we will work on, I guess, how the view. We did note that part of your presentation is cut off by the faces, so we'll look at that for next time. So thank you. It's great. Oh, here's Hans again.
Thanks, Mickey. A colleague once told me hope is not a planning tool. And so as we transition across to Dr. Nancy Shellhorn, I hope that this will be a smooth transition. So I believe she's joining us today from Brisbane. Over to you.
Hello, Hans. Can you hear me okay?
Yes, clear and clear.
Fantastic. And can you see my screen okay with the presentation?
Yes, we can. Thank you, Nancy.
Fantastic. Thank you. So thank you so much, Hans, for the invitation, and a good afternoon to the participants. I'm sorry that I can't be in the beautiful Coonawarra. I wish I could, but we'll do our best here from Brisbane. So I'm Nancy from RapidAim. We provide pest forecast news and alerts. We basically help farmers reduce the risk of loss and cost from insect pest. As most of viticulture producers would know, you use insecticide as a means to kill pests, but mostly as insurance, because you can't take the risk, and it's kind of just in case a pest is there. So for example, light brown apple moth. So, and that's because pest management's a guessing game, and you often don't know when and where the pest shows up. And that's what RapidAim, the problem that RapidAim solves. So we're a digital crop protection, and it's really, we put this information in the hands of growers so that you know where and when pests show up, so that you can target management and then validate whether control is working. So I'll tell you a little bit about our story. So we're former CSR scientists who were really years and years of working on integrated pest management in a range of commodity crops. So everything from cotton and grains to vegetables and fruit. And often the biggest barrier was growers just, it's risk management. So we started to develop some technology that would allow us to know when pests show up. So we decided to exit our very comfortable jobs at Sorrow. We formed a company and we raised capital. And our first application is fruit fly. Now I know that viticulture, it's a very, very minor concern, but I think the exciting part is that now that we're in the marketplace with over 500 devices and 170 end users, we're starting to add additional insect layers. So for example, light brown apple moth is one we're actively working on. So I'll tell you a bit about our technology. So it is a hardware enabled software system. So I guess since you can see me, these are our devices. This is our first, it's a fully integrated IoT device, super simple to use, plug and play. This is it. There's no additional big solar panels or star pickets. Really, customers, they sign up. We ship them a grid of, they start with a grid of 10. It's meant to be used as a grid so that you can get real-time estimates of pest populations on your farm. They download the app, they scan the QR code, and basically they just, open it up, plug in the battery, and the whole thing goes live. So super simple to use and fully integrated. And then it communicates, there's an attractant or lure inside the device, just like there would be for any manual device. And but our novel sensor, it's ultra low power, and it's kind of like the behavioral fingerprint that you use to unlock your phone for your fingerprint ID. So it detects, and then the algorithms that sit with that sensor discriminate the insect that's coming to the trap. And then that's some edge computing, so onboard computing in our little tech pack. And then that's sent to the cloud. It communicates by narrowband IoT, which is meant to be for ultra low power bandwidth of communication. And then that goes straight to the mobile app of the end user. And you can see here that's the interface. So that's our piece of hardware, and it's quite durable. So we do surveillance, for example, for biosecurity for fruit fly. These devices are meant to be out in the field for years, super low maintenance. They do have to change the lure about once every 5 months. They're made out of same material that headlight covers are made out of. And then the real product though, so that's just our infrastructure, the sensing devices. The real product is the information that growers get in real time in a mobile app. So it's like an on-farm pest insurance app. And you can see here, growers will then know where the pests are and when they're active. So these are real examples of some of our customers who have spread the sensors out across their farms. The granularity of our information is such that they can see the time of day that pests are active. And you can see in this example exactly when on the 24th of March, fruit flies started waking up on this farm and were most active. This has completely changed farmer behavior in terms of applying insecticide. A lot of growers, often, particularly in warmer climates, won't apply product until late at night. But particularly if they're using a knockdown, this means that it's not very effective. But by seeing when they show up and also on farm, this allows for better targeting. Here's another way that the product's being used. Oftentimes, growers, producers have multiple farms, and just by opening up the app, you can see which farm might need attention first. And you can see this one down here certainly has a hotter, getting more detections. When you click on those, you can open up the sensors across that particular farm, and you can actually see where there's a bit more activity. So it helps to prioritize workflow. And you can see that action was taken here. And then 2 days later, they can see whether or not they're getting a level of control that's acceptable. So in addition to the on-farm sensors, we're also adding value to this real-time data flow. And we're starting to build pest forecast. So the product now always shows you what's happened in the past. But now, of course, because we have this highly granular real-time data flow, we can start to project into the future, ultimately to help growers plan and prepare, and to know how close the issue is to their property without actually divulging who has a bigger pest problem in the region. So our technology, like I said, we came out of CSRO, and we validated the technology with the federal government with a project back in 2019. And so we deployed our first technology, and that's where we used, you've probably maybe heard of LoRaWAN technology, where we had gateways. So 5 regions, 290 sensors, 55 end users. And we did this for 6 months. And back then, even with our first product, in terms of the reliability of our product, we got about a 2% false alarm rate. So about 2% of the time that overcounts, and we rarely ever missed a detection. But most importantly is we showed that the huge cost savings from not having to go around and visit traps, and particularly if there's an outbreak for fruit fly, the cost that's involved there. Because this is early detections in real time, they can respond to it so quickly and really define area freedom very quickly. But I know this is fruit fly, but you can imagine for things like light brown apple moth, or even for for some of the other pests that might be of critical importance, earliness and real-time alerts, and then the location where they occur allows for a super tight link with management. But I think in terms of the future, because I think we really are, digital crop protection and biologicals will become more and more the future of pest management, and less reliance and dependency on chemical. You can start to imagine that with the combination of Rapid Aim, where we're providing a forecast, it allows growers to plan and prepare. And then you can use a range of products to protect your crops, whether it be proteins, biologicals, even targeted chemicals. But then the thing that I think is most exciting, and certainly what our customers tell us they love about Rapid Aim, is it proves they can see whether or not the product and their management is working. So it gives them that confidence piece. So who are some of our customers and the kind of things they say? They talk about that it's the future, that it definitely, if Having information means they can take action now and reduce a bigger problem later. It also means a change of behavior and a change of practice because of that, strong need to manage risk, moving away from the idea that you're just having to spray because you can't take the risk. So really knowing where the problem is, knowing which vineyard the problem is existing, and then being able to target your management action and knowing whether you're getting control. So what have we done so far? Well, we became fully commercial with our first product back in January of 2020, and we now have over 500 sensors across Australia and New Zealand, and the mobile app in the hand of about 170 end users. We're super cost competitive compared to manual monitoring, and it's basically about $366 to lease our device and get the information per year. So it's about $1 a day for a 12-month contract. We highly recommend that they're rolled out as grids, because the power of putting multiple sensors in your orchards, on your farm, means that you have a granularity of data and a level of confidence that's much, much higher than if you just put out one or two. If you only put out one or two, what you get is a trend over time. So not a very tight link with management. But when you put out a grid of sensors, then that's where it's super powerful. The way we've gone to market so far, though, is that we roll out our technology across entire regions. So for example, we have about 155 sensors rolled out across the Yarra Valley. And so that's for surveillance. We have other grids rolled out across the New South Wales coast. So those large scale grids really allow us to provide the forecasting component and that early warning. And then growers in the region purchase more sensors for their own property so that they can actually see not only the information we provide them across the region, what's happening as a forecast and as news, but actual real space and time information of the insect pest in their orchards and in their farms. So yeah, that's Rapidae. We're taking the guesswork out of pest management, and I welcome any questions. So thank you very much.
Great, thanks, Nancy.
Right, now everybody in the Limestone Coast is back. Right, so I'm here to present to you once again. Kath Kidman's not here, so it's me, but really we've worked on this talk together. And it's to do with a project that we've done as part of the Regional Partners Program for the last 2 seasons, looking at the impact of virus in the Limestone Coast. So first of all, I wanted to highlight this picture that was posted on social media through our Limestone Coast Facebook page. And it's a photo of, that's Panola down here, and that's the Terra Rossa strip heading, the Riddock Highway heading north towards Narracourt. I suspect this photo was taken probably in early April, so most of our grapes hadn't been picked in the region then, and you can see how much red we are dealing with in this particular region this year. So just wanted to emphasise, when I saw this photo, I was horrified. I don't know for other people in the region what they thought. Certainly a lot of the commentary thought how beautiful it was, but for me, that's horrific. So anyway, I just wanted to emphasise, I certainly don't know the answers to the issues that we're dealing with and what I'm going to speak to today. And certainly some of the questions that play through Kath and my minds when we're walking our vineyards is, what role does virus play in preventing our vineyards from achieving yield and quality? I know Fiona put out some stats there from work that's been done. A lot of it's been done overseas, so it's not that relevant to what's going on here in Australia. So, and a lot of what we talk about is anecdotal. We don't really have numbers against it. So we'd like to get a better understanding of that. And what proportion of our vineyards are infected with virus? You know, obviously you look at that photo, you'd have to say a large proportion of this region would have some form of virus in it. Are some wine regions worse affected than others? Can't answer that question. We've certainly walked a lot of the regions this year for Treasury, and it's in every region. It's not just the Limestone Coast, and I suspect Margaret River would comment to the same effect as well. So does climatic variation play a role? Does that have something to do with the regions that we're talking about? Are there varietal differences? And I would certainly think that there are. But we need to understand, how does this virus spread and what role do those vectors play in the virus spread? So, you know, I'll talk a little bit about that, and obviously other speakers have done that today too. I'm not going to run through these in any detail because it's really been done by previous speakers. Just to let you know, though, they're the main viruses that we deal with on a day-to-day basis. We need to understand that obviously we can transmit those viruses through propagation material and grafting, but obviously most of them, other than leaf roll 2, and we've got a question mark there because maybe it can be, can all be transmitted by vectors as well. And once again, what is acceptable to industry? I think this is the $50 million question. You've certainly got winemakers that would think that things around GV, like leaf roll 9, where we know we talked about SA125 as a clone in Cabernet, being acceptable to have a bit of virus in it because, you know, it gives us the quality attributes potentially that they're after. Is that true? I think a lot of it's anecdotal. So, you know, and lots been moved on from that work that was done back in, I don't know, was it the 1980s, 90s around SA125 and SA126. I'm not going to go into any detail with that. But I would have thought, as a general rule, we wouldn't be accepting any virus material coming out of a rootstock. And I know that there's certain people that would say that rootstocks don't carry virus. Well, I'd beg to differ. So these are photos that Kath and I have taken over the season, and I'd really like to move my photo away from that grapevine, the bunch. Is that possible? Oh, I can. I go like that? Yeah. So, oh, it's still up there though.
Just do it. You can do it over here.
Oh.
Do it something like that.
All right, perfect. Okay. So the mealybugs that we're dealing with in this region are these little white ones here. Well, actually, they're a little bit orange. So these are the, let me get my notes because once again, I do not claim to be. These are the subterranean, the obscure mealybugs that we're talking about here. So why they're obscure? They like to live underground. So, which is different from the mealybugs that they were talking about in New Zealand. So be aware, these are roots from a vineyard not very far away from here. This is bunches of grapes. Oh, shivers. How do I go back on this? So, yeah, these are being found locally. We've had them tested and you can squeeze them and they turn orange. So that tells you straight away that that's that particular mealybug. And obviously scale, we have, this is actually a really young vine, as you can appreciate. It's only just got up onto the wire. But you can see those little scales there. So, you know, they're the mealybug, sorry, they're the vectors that we're dealing with down here. And they really do pose, and we've heard from previous speakers about the issues around those. I don't think we really understand the impact. We don't actually know how much we've got. We don't even know how to monitor for these things. So I think we need to get a better understanding on all of that. So how are we going to prevent further spread of virus and protect the longevity and quality of our new vineyards? And so this is where I move into our project, and I've called our new vineyards our treasures, and I play on it, the words of our promotional material for our region. So in 2020, we undertook a virus survey across 6 vineyards in the Limestone Coast, 2 from Padthaway, 2 from Rattenbully and 2 from Coonawarra. We asked for a vineyard of a white variety and a red variety, and we wanted vineyards that were under 20 years of age. We did the surveying in late March, April 2020, which you can all recall was, you know, when we all shut down. So it was a really challenging time to get this done. So you can see there that we had a Shiraz vineyard, we had 1 Chardonnay vineyard that was on a rootstock, we had 2 Cabernet vineyards, and we had 2 Pinot Gris vineyards, and that probably is reflective really of what we've been planting in the last 20 years into our vineyards. So straight away, that gives you an idea of what vineyards we targeted. We then created maps like this. So we were surveying these vineyards where we were taking a 200 vine composite sample across a block. And obviously what I listed before, those 6 vineyards, that each one is considered a block. We then had composite of samples. We had to, as you can appreciate, to do 40 samples by $100 a sample, each vineyard to test was over that $4,000 mark. So to test using PCR to get the numbers that we wanted is quite expensive, hence why we could only do 6 vineyards really intensively like we've done. So when I talk about a composite sample, I think this is really important so you understand what we're dealing with here. See this map here, and I'll use this number 1 here. That is a 5-vine composite sample. So 3 leaves were collected from this vine, 3 from this vine, 3, 3, 3. So each of those 3 leaves were put into a separate bag, but then that went into the lab as 5 separate bags into 1 bag as a whole. The lab then analysed that bag. They took 1 leaf out of each of those 5 bags, took a little sample out of it, and then they blended them all up and put them through the PCR machine as one. So the results we get back are reflective of those 5 vines. And in this case, you can see in this row 7 and 8 here have tested positive for leaf roll 1 or leaf roll 3. So we then obviously put all that information together and pulled these maps up for each of the vineyards in question. This particular vineyard, and we then broke down, because obviously we're talking about a lot of viruses that we're trying to tell a story around. We obviously understand that leaf roll 1, leaf roll 3 are the baddies. They're the ones that we really do not want in our vineyards. So we've made those and called them high severity. That, you know, if you've got them in your vineyard, you've got a problem. We then class the moderate severity as being viruses that were complex, such as leaf roll 9 and GVA together. You know, lesser priority, but it's still a virus and it could have problems if it complexes together. And then obviously the green in this case is the lower severity, which might include leaf roll 4, 5, 9, GVA or GVB if found in isolation. So we really just wanted to tell the story when we presented a map to these particular growers on how bad those severity compositions were. If you were this vineyard, you'd be a little bit worried. So in summary though, I'll give you an idea of what we're dealing with. So if we look at vineyard leaf roll 1, out of the 6 vineyards, we had 2 that came back positive for leaf roll 1. We had none that came back for leaf roll 2, which I would hope would be the case. Leaf roll 3, 4 of the 6 vineyards had leaf roll 3, only 2 had leaf roll 4, 2 had, I didn't highlight that one in yellow, 2 leaf roll 5, and then we had 4 with leaf roll 9. None with the Grapevine Pinot Gris, which is nice. We had 4 with GVA, we had all of them with GVB, and we had them all with Rupestre stem pitting, which concurs with what we've heard earlier today. I would suggest, and I'm going to stick my neck out here, this is very reflective of what we would see across the Limestone Coast. You know, we pulled 6 vineyards out, but I would say in the young vineyards case in this region, this would be a very true story. And I can obviously comment on that because we see a lot of other young vineyards across the region. And I just want to let you know, none of these are treasury vineyards. These are growers. So just to let you know. So just be conscious of those numbers. And then, well, now I need to move that back. Other mouse. Move it back that way. Can I do that? Right. Oh, why does that not move for me?
Because we've got 2.
I'm really
rooms.
confused.
This is the green room. So,
Oh.
so that, look up there. And where do you want it? Back up there.
Oh, just move it out of the way of the table. Yeah. Oh.
Over here.
Yeah, that'll do. Yeah. Okay.
Yeah.
So now just remember those categories of high, moderate, and low. So in Vineyard 1, we had low severity 100% of the time. And if you remember Vineyard 1, that was Shiraz on a replant situation. Vineyard 2 is a Chardonnay block, rootstock. Half of it had high severity, the other half low severity. Obviously, they can't see the visual symptoms in that block. This was a replant block. It's probably getting close to 20 years of age. It was planted in the early 2000s. Vineyard 3, 30% of it was high severity, 63% moderate, 7% low. This vineyard is a replant situation and obviously has issues. Vineyard 4, very low severity, 100%. That was a Pinot Gris vineyard. Vineyard 5, high severity, 21%, moderate, 79%, and obviously had no low severity. That vineyard's in trouble. Vineyard 6. Now, Vineyard 6 is interesting. This is a Pinot Gris vineyard. It's got a high severity 13% of the time, but it was a virgin country. So you would have to assume that this particular vineyard has received that virus load from planting material. So in all other cases, it's a replant situation. So you'd have to assume that that's maybe a legacy issue in a lot of those cases. You can't always assume. It could also be planting material. And I can say Vineyard 3 is certainly planting material. So, you know, you really have to delve into this information when you start to make big assumptions, because there could be a lot going on. So that was 2020. And 2020 was challenging because the labs, to get the turnaround time to get the answers was really late, because I don't know if you realise the PCR tests that they use are the same as what COVID uses. So, you know, it was quite complicated to get our samples done. So in 2021, we went back to Vineyards 3 and 5. Surprise, surprise, they're the two Cabernet vineyards, because we decided that it was worth investigating a little bit further some of the questions we had arisen. So basically the retest strategy included revisiting the vines that we tested in 2020. We then went and tested the vine next door to see if that had any positive results, even though it might have still just looked green. We wanted to look at nutritional status and really eliminate that as an issue. We also compared labs. We sent paired samples from the same cane of vines and sent them to 2 different labs. So I'll show you some of the results of that. And then we remapped the VSI symptoms that we saw to give you an idea of increase. So the first picture I wanted to show you there, this particular vineyard, that this is 2020, that vine, and this is in 2021. This vine came back positive for, came back positive for leaf roll 3. So obviously it's nice to think, oh, you could go back to that vine and still see that it's exhibiting the same symptoms, basically. That wasn't always the case. Right, the next one that I'm showing you is testing the vine next door. So these are some young vineyards that only be a couple of years in the ground. This particular vine here, you can see classic leaf roll 3 symptoms, which it came back positive for leaf roll 3, 4, 9 and GVA and rupescent stem pitting. But the vine right next door to the north of it, it didn't have leaf roll 3, and as you can see, it's just green. But what's to say that next year we come back and it might be red, because we do know there's vector issues in here too. Now, apologies for the photo. It was drizzly the day that this vineyard was walked, but this is around nutrition. And we did this on 3 separate vineyards. I've only put the one up, but we saw the same trends. And you can see here where the green vine is green and red is virus. You can see the virus status of those vines. The one that's red obviously had leaf roll 3 versus the green only had leaf roll 9. But you can see straight away that we've got some really big differences in the magnesium numbers, calcium, manganese, molybdenum, phosphorus, potassium, zinc. So straight away, I would assume if you think about a red vine, they are not going to be photosynthesizing to the same capacity as that healthier green vine. So obviously they only turn red at veraison traditionally, or, you know, after veraison. So the ability of those vines to take nutrients, use them properly, is obviously going to be compromised. And over time, you would think that these numbers will only get worse. So, and that trend was consistent across 3 different vineyards. We saw very much lower, and you know, phosphorus in this case isn't much lower, but in other vineyards, phosphorus and potassium were the 2 that really stuck out. Now, labs, comparing the labs. In the case of the one on the left, that vine, now I've got lab 1, lab 2, and then I've also got lab 2 because, of course, we tested these vines back last year as well. So lab 1 has tested this vine and it's come up positive for leaf roll 3, 9, GVA and rupescent stem pitting, whereas lab 2 is actually telling us that it's got leaf roll 2 and not leaf roll 3. It's got leaf roll 9, GVA, rupescent stem pitting. And then obviously back in last year, lab 2 did tell us that that had leaf roll 3. So we're making some very big decisions on these test results that we get back from the labs. So I just want to pose a question here, and I'm very happy to talk to Fiona around this, and we do need to have some off conversation discussions around this, but it's not, I've put another example up here. Generally, it seems this one is a lot, we've got leaf roll 1 confirmed for that vine and leaf roll 9, but, we've got one telling us it's got GVA and the other one not. So just to get some consistency, I suppose, around laboratories, it could be our sampling methodology as well, so I won't discredit that, but we've tried to get the leaves off the same cane, but then Kath wanted me to pose the question, traditionally virus testing used to be you take wood cuttings, send them off and grind. These are leaf samples. Is the leaf that we're selecting maybe not the right leaf to, that's all the sort of stuff that we don't know, and I think we need to really clarify, because as I said, we make some very big calls on these results that we get back from the laboratories. And then finally, this last one is around when we went back out and remapped these vineyards. You can see here, vineyard 3, we went from only having 40 visually symptomatic vines last year up to 110. And in vineyard 5, it went from 52 up to 162 or something like that. So that's like a 3 times increase in the number of vines that have turned red in one season. And I would have thought what we can see across this region, it'd be interesting to know what Margaret River's saying, but that increase doesn't surprise me if we compare just last year to this year. So I would have thought, and obviously we've got a lot of other numbers from our company stuff as well, but for the purposes of this talk, we stick with the survey. So just moving away from the survey, that's the results. They will be written up. We've written up last year's results. We've obviously just waiting on some final test results to come in from one lab. But basically, when we look at our focus areas going forward, as I said, we make some very big decisions. When we look at the results of our testing, we obviously do a lot of virus monitoring. We do a lot of virus testing. We have big questions about roguing and replanting. We don't know if that's the best way to go. Vector monitoring, vector control, I'd love to think we can get a lot better at it. I don't think we know very much at all at this stage. And obviously then vineyard hygiene. So we also need to think about our source blocks. So if any of you have source blocks for planting material, really need to be how well are we monitoring those? And that goes back to a nursery situation as well. I would suggest it's probably not good enough. Blocks destined for removal, how do we treat them versus how do we treat a newly established vineyard that's less than 5 years in age, and then how do we treat established vineyards? We've obviously got probably different methodologies around how we treat those vineyards. So can't emphasise enough, this is obviously a vineyard that's been recently ripped out, and you can see the root material that we're dealing with there. We need to be very conscious of testing for legacy, which is what we call it. We would be testing these vineyards before they get pulled out so that we understand what virus load that we're potentially dealing with. So can't emphasise that enough. Obviously, the visual symptom identification, this is more maps around what we've been doing when we map our own company vineyards there. It's really time consuming. I'd love to think the technology that's been shown today becomes commonplace, because I think it's only really good for red varieties too. So we need to take that into account. And can we distinguish all strains of leaf roll via VSI? Obviously, I think I'm confident enough between 1 and 3, but I don't know if we could do that for the other strains. And it's very limited to when we can actually perform this walking. Obviously, it's post-forays, and it's usually the later you leave it, the better. You'll get a more accurate count. But obviously that's at a very busy time of the year, and you'd love to beat the harvesters to when you do it as well. And you do need some training. You can't expect any Joe Blow to go in there that's got no idea about what virus is or what it does. So you do need some training there. And obviously you can't be expected to test every vine either. That's the issue. So that economic issue as well. And then obviously that question I posed earlier, that tolerance, how much are you willing to tolerate in your vineyards as well? And that's something I don't think we've got a good handle on either. So our current monitoring for protocol for the young vineyards, so these are the ones that would be under 5 years of age, is we would be doing an annual VSI program, so walking every row, and tagging, mapping and roguing those vines annually. But as I said, we are starting to question the roguing, whether the feasibility, especially if you're going to go in and replant straight after. I think we're learning that that legacy issue is very real. And obviously getting a better handle on our vectors and really understanding what we're dealing with. In our older vineyards, we would need to think about establishing, getting an idea of what that virus status is in those vineyards, and actually then zoning them out into high, medium and low, and I'll show you another slide in a minute, and then obviously looking at that vector status as part of that virus zone. And it's probably putting them into a hold pattern and trying to ensure that we're not increasing that virus load any higher in those vineyards. And this is just how the risk status is done at the moment. We would have a low risk, that would be where there's no virus and or low no vectors. Then you can have two categories of moderate risk where there might be no virus, but you know you've got the vector. Or the other one is you've got virus, but with very little or no vector there. And then obviously high risk vineyards. So you would try and zone those vineyards into the same areas, and then manage accordingly, you know, disinfect equipment from those zones to try and minimise the potential spread of your vectors. So into the future, we're hoping that we can understand the role of virus in our vineyards in order to protect the future, and that includes our heritage propagation material, which obviously we do a lot of. How do we maintain the longevity of existing old vineyards and secure the future for newly planted vineyards? And in regions like here, which is different to Margaret River, I know, but we've obviously got vineyard after vineyard that are just all slammed next door to each other, not so much like Margaret River where they're a lot more spread out. So when you're trying to manage it, it needs to be a whole regional action plan. It can't just be one person trying to do it, because you're going to be fighting an uphill battle. We need a lot more research into virus in Australia. We cannot rely on overseas data to a lot of, we cannot rely on overseas information because it may not be relevant to our climatic conditions. And I think we're seeing that with what Vaughan's presented this morning to what we see here. Obviously, we're dealing with different varieties straight away, really. So then we need to develop targeted management strategies for leaf roll vineyards. And we'd love to think that we could obtain similar levels to what Vaughan was talking around that 1%, because at the moment it's a lot higher than that. So I think I just wanted to say thank you. Obviously, a fair bit of money has gone into this from Wine Australia's regional program. Obviously, thanks to Uli and the council, to the vineyard managers. Obviously, we don't name them, but thank you very much to the 6 properties that provided sites, obviously the labs as well. And there will be a final report, and it will go up on our website once Kath and I have finished writing it. So thank you.
Yeah, thank you, Kerry. Fantastic. That was good stuff. Andy Gordon, you're next. Are you online? I'll read the bio for you. And yeah, it's next. Okay, so Andy Gordon, KC Vines and Chair of VINA 2021. Andy was educated at Melbourne High School and Melbourne University for physical education. He taught, coached and lectured for 27 years and has wide business experience in high volume food retailing, commercial health fitness, property development and elite level sports coaching, swimming and basketball. Andy has been a member of VINA since 1998 and secretary since 2004. Andy is the proprietor of KC Vines and Rootstocks, a wholesaler production grapevine and almond tree nursery at Trentham Cliffs on the New South Wales side of the Murray River at Mildura in Victoria, northwest Victoria. Andy bought the Trentham Cliffs property in 1998, established the nursery. The nursery boasts 7 greenhouses with 350k capacity, 2 hard stand areas and a capacity of 950k field planted grapevines and fruit trees.
Okay, I guess that's over to me, is it?
Yes, over to you, Andy.
Okay, all right. Well, thanks for that. Thank you for the invitation to make a presentation to this group. I've been sitting here with a lot of work to get done and trying to work away with the proceedings in the background, which look, what I did garner from the proceedings was, very, very interesting from our point of view. Some aspects of it particularly interesting, which was the testing and so on, which is really germane to what the nurseries are on about, to make sure that any problems with the health status of material coming out of nurseries or coming into vineyards is minimised by the activities of the nurseries and the supplies of propagation material to the nurseries. The state of play at the moment with regard to propagation of planting material, there are two parts to this process. The first part is the supply of propagation material to the nurseries. Now, the sources of propagation material that are going to the nurseries either come from private source areas or they come from the vine improvement groups. And nurseries also do some collection of vinifera and things like that. But I guess I'm talking specifically about rootstocks with regard to the commercial source areas. And the second part of that is the processing, maintenance and nurture and delivery of the propagules to the client customers. Now, the nurseries themselves are actually the middlemen in the whole process. Most of their material, apart from those nurseries that have their own private source areas, most of their material is brought in. And what comes in with that material is whatever particular pathogens or characteristics that might be a part of that material that comes from the source area. So the nurseries work with what they're given, and then they obviously go ahead and do the propagation, look after them and so on, and then deliver them. So the state of play at the moment is, and I've got a series of dot points here and I'll just read through them. The growers or the end users have no guarantee that the planting material being supplied to them from the nurseries is disease-free, as well as the nurseries having no guarantee that the propagation material being supplied to them is disease-free, and that a legally binding guarantee is probably unachievable given the nature of the business. These dot points are reasonably random. There is no national standard for the health of planting material in Australia. There's no agreed national certified standard. Rupestris stem pitting is endemic around the world and as such is regarded as being acceptable. Not that there's much that we can do about it at this stage. Source areas for nurseries are not registered or audited for disease testing. Source areas, both vine improvement and private source areas, are periodically tested for virus infection. There is no agreement on what constitutes healthy propagation material. When I say agreement, this is general acceptance across industry or something that's been formalised. There is no Australia-wide or industry-wide agreement. Nursery source propagation material from vine improvement associations and other commercial source areas and private vineyards. Some vine improvement associations virus test regularly, and I believe most of the vine improvement associations are pretty good at undertaking virus testing on the material that they're supplying to nurseries. Virus testing is expensive, and there are limitations on how much testing can be done. And I found it most interesting with one of the presenters talking about the new technology available or becoming available for virus testing. That is of particular significance to nurseries. There is no agreed protocol for virus testing of propagation material. I mean, how many vines per 100 vines in a source area should be tested to give a reliable indication of the health status of propagation material being tested? So this is one of the points that in setting a certified standard has to be dealt with, and if some agreement has to be reached. Some nurseries claim their vines come from certified propagation material. Now, this is a furphy, because there is no certified standard and there is no regulatory or auditing body in Australia. So when when nurseries are claiming that their material is certified, it's not certified to any agreed standard across industry. It is really just a marketing ploy. A lot of the high volume nurseries have hot water treatment plants in, but as we all know, hot water treatment doesn't kill viruses. It's in there generally for agrobacterium and for nematodes and phylloxera. Hot water treatment does reduce the titer of some trunk diseases. It does kill nematodes and phylloxera, and it does kill agrobacterium, but not 100%. There is, when material has been hot water treated for at 50 degrees for 30 minutes and then subsequently tested, then there are traces of trunk disease and agrobacterium found, but it does reduce the titer enormously and reduce the risk. VINA was set up by the vine nursery industry to give it a body that could address some of the issues concerning the rapid planting of vineyards in the 90s. There was a lot of concern about where the material was coming from, what its health status was, and there was a frenzy of planting and a great shortage of material, and vineyard owners and managers were inclined to take whatever they could get to plant their vineyards. Now, this eventually gave rise to the Vine Industry Nursery Accreditation Scheme. Prior to VINA's existence, there was no organisation for setting standards in the vine nursery industry. So the accreditation scheme was established originally by Avia, and then subsequently VINA introduced their own. Now, Avia operated for a couple of years. I know our first, as a nursery, we were audited and accredited by Avia, and then later on we transferred over to the VINA scheme. Quite often there's confusion about the two terms of accreditation and certification. Accreditation is not certification. Accreditation is about an audited production process. Certification is about certifying the health status of propagation and planting material, and these two terms are not interchangeable. VINA has been concerned for a long time about the lack of a standard. And so 3 or 4 years ago, the VINA committee resolved to work towards setting a VINA certified standard. Now, the nurseries felt that they are the appropriate body through VINA to set a certified standard, because they are the ones that deal with both the supply point and the end user point, and the nurseries are the ones that have to answer to the clients about any deficiencies in the planting material that they're supplying. So when you look at the state of play as being described here, and you consider, well, where to and what can the nurseries do to lift their game or lift the whole standard? And it seemed with the VINA accreditation scheme in place, it would be a reasonably simple matter to implement a certified standard once agreement with all the stakeholders in industry had been achieved about all the various factors that would come into setting a certified standard. Once this has been achieved, it's a very simple matter to slip that into the existing accreditation scheme. VINA already audits a number of vine improvements under the VINA accreditation scheme and a number of nurseries. The nurseries are generally the high volume nurseries that do bench grafting. There are not a lot of field grafting nurseries that are accredited. Some are members and some do their own thing and don't want to know. One of the steps that we are taking to get this certified standard up and running, we've devised a bit of a roadmap, a way forward, and we have a working group of experienced nurserymen who are working on gathering information and putting something in place for industry to consider. We are looking for funding. We're busy people, and we're not able to take a person from our staff to work as a project worker under this. And we need to get some funding for a project worker to work under the direction of the VINA committee to survey existing certification schemes around the world to identify commonalities, to establish a register of commercial source areas. Now, that's a source area that sells production material, liaise with industry on setting the standard, to revise and expand the VINA accreditation scheme to include the certification scheme. The certification scheme, as I said, fits very neatly into the existing scheme and its auditing structure. Plan and execute a program to promote with growers specifying the use of propagation material that firstly comes from a VINA accredited nursery, and then secondly, once this is in place, a VINA meets the VINA certified standard. So a number of criteria have been set for people or nurseries that want to use the meets the VINA certified standard tag. There have been criteria put in place. Firstly, their source area has to be registered. The source area is where the material is coming from, has to be registered. Secondly, they've got to be VINA accredited. And then thirdly, they are subjected to annual audits whereby all their virus testing results are perused and signed off on, and then permissions given to use the meets the VINA certified standard is then passed on, and they are able to use that tag as a descriptor. This then enables industry, once this is in place, this enables industry then to specify the material that they want to plant in their vineyards, whether it's greenfield or whether it's replants. That material is as good as it gets when it comes to viral health status, and we would be hoping in the future the status of trunk disease and other pathogens. So we have developed a roadmap. We have a way forward, and we're working towards that. And there are a number of hurdles or a number of things that we have to resolve. But one aspect of these are the issues. First of all, to meet the VINA certified standard, material obviously has to be true to type. So that will require DNA verification. And with a bit of luck in the future, AWRI are hoping to run one that will enable RNA examination to identify clones, which would be another really good step in the right direction. To determine with all the stakeholders which viral pathogens, the decisions of what's in and what's out, and also what method of testing is the desired method of testing. Some areas use ELISA, most use PCR. And with regard to sample testing, the frequency of sampling, the interval between sampling every year, every 2 years, the level of sampling, how many vines per 100, the site of sampling on the vine, whether it's distal, proximal canes, etc., the time of the growth cycle for sampling, which month is best. These are the protocols that need to be sorted out as part of any certified standard. And obviously, suppliers of propagation material would need to be members of VENA and accredited, and so on. This project is the biggest project that VENA has tackled since the implementation of the accreditation scheme, and it'll take time to implement. With regard to that, let's assume that a certification scheme gets up, and we have, we've answered all of these questions. So the next thing is, well, where to from here? Now, to get source areas that are all going to have material available, both rootstock and vinifera, that's going to meet the certified standards, not going to happen overnight. There's just not the material around. So it would be a process of roguing out and replanting, well, testing firstly, roguing out and replanting. And that's going to take a while, and there would be a transitional period, one would have thought, of 6 to 10 years before all of the registered source areas meet all the criteria for certification. However, that doesn't stop the end users, when they're ordering material from nurseries, to specify that A, it comes from an accredited nursery, and B, that the material has been or hasn't been tested. They need to find out what the status of that material is. VENA is working on, in addition to this, some protocols for our members with regard to being transparent with end users, so that they are in the driver's seat when it comes to making decisions about accepting or not accepting material of a particular health status. So that's reasonably important, and I would have thought professional organisations would embrace that sort of approach.
Well, thank you,
and by way of a bit of preparation for this presentation, I spoke to Alan Little at AWRI just to get some statistics on what the level of testing is at the moment. And I will read to you. He's answered my questions as best he can. My questions were probably didn't suit the statistics that they have available. So the total number of grapevine test requests in 2020, can't provide an exact number, but the test requests we received, 77% were from vine improvement or nurseries, 6% from agronomists or viticulturists, and 16% from commercial vineyards. 99% of the requests are for PCR. They rarely get tests for ELISA. The number of PCR tests requesting the full range of testing, including Pinot Gris virus, was about 50%. They didn't keep a number on the full range of tests that didn't include Pinot Gris virus. The number of PCR tests requested with leaf roll 1, 3, GVA, GBB without Pinot Gris, they package up their testing in such a way that that wasn't really relevant. They offered the 3 virus pre-graft screens of leaf roll 1, 3 and GVA, and 23% chose that. A 4 virus pre-graft of leaf roll 1, 3 and GVA, 5% went for that.
Thank you, Andy. in interest of time, we're a little bit behind schedule. They're all really good points that you make, and it's good that we're leading into the Q&A section of what we're doing. So we could pick up some of those ideas as we go into the next session. So thank you for your presentation. It was really good. Thank you.
My pleasure.
I'll get Hans to come up and I guess run through, well, I won't call them rules, but the way we're going to run the Q&A. And have you got some questions? Yes, you have, and there might be some from Margaret River as well. So, but I'll hand over to Hans, and yeah.
Thanks, Ollie. I'd just like to welcome back into the room Dr. Fiona Constable, Dr. Vaughan Bell, and Dr. Monica Keogh. You'll see them appear shortly. I'll also introduce Chris Rogers, who's here with us in Coonawarra, who'll be on the panel as well. Chris grew up on his family vineyard property in Eden Valley and has over 30 years experience in Barossa viticulture across technical, vineyard management and grower liaison roles. He is a Charles Sturt University graduate and has formal training in winery operations, compliance systems and team leadership. Chris started his own viticultural consultancy business in 2018 and provides services to growers, wineries and regional associations across Barossa, Clare Valley and Adelaide Hills. Chris has also worked at Yalumba Nursery and has been involved with Barossa Vine Improvement since 2004 and is the organisation's current chair. Barossa Vine Improvement is a not-for-profit organisation established in 1967 to provide accredited vine cuttings to growers and nurseries for new vineyard plantings and grafting. Barossa Vine Improvement is also active in new clonal development and works closely with the South Australian Vine Improvement Association, viner and industry researchers to advance the need for high health planting material and superior clones. Introducing Chris on the screen. And just to get the session started, just a quick question on your thoughts and any thoughts that you have on vine virus in your interactions with growers, grower clients, through to industry organisations that you're involved with.
Thanks, Hans. Welcome everybody. Welcome to Cohort City, University of Australia and elsewhere. I made a few notes. I guess the first thing I would say, huge hats off to the organisers. This is certainly one of the most comprehensive gatherings I've seen on grapevine virus and associated issues. You can imagine I've done a lot of reading, talked to a lot of people in my time, and obviously been to lots of seminars, webinars, remind people of some really great resources online, they do it right, and so forth. So huge hats off, really well done to bring it together. As I said, this is a really good collective of knowledge. So many things, whilst I guess I don't pretend to know everything, there's a lot of familiarity with what's been presented here today. A lot of really, really powerful take-home messages. Hats off to Wine Australia for not only funding this, but also just for your knowledge, there's a lot of activity going on in the background with regards to Wine Australia's involvement, really ramped up in recent years to continue to advance what we need to do in this space. There's a lot of people involved in this presentation today are involved in the background with projects, developing best practice, best management practices. There's a lot happening in the propagation and vine improvement sector about developing their standards. Certainly in that front, I guess for me, with my experience both in management, propagation, working with clients across multiple regions, I'll probably break it into two parts. And one is in regards to established commercial vineyards, a lot of what we've heard about today, and obviously the dealing with existing virus, reducing the risk of virus incursions and spread in those vineyards. And then the second piece, I guess, is where I'm also involved in the propagation sector, as is Andy and other people in this room and on the telecast today. So we have a lot of things in common. A lot of the technologies being presented today are common to both of our needs. I certainly would, again, highlight that call out for more research, absolutely essential. There's a lot we know, but there's a lot we don't know, especially regarding testing methodologies. And just, I guess, on a practical note, with regards to the testing, Vine Improvement does a massive amount, 77%. We spend tens of thousands of dollars in virus tests. If some people are going in doing molecular testing, don't just rely on one result. In our sector, we would probably do 3 before we would make a call on what we do in that source block, that being where would cuttings come from. And that's not to question the testing, the lab, that's the question sampling methodology, the timing when you take that sample. So we've seen all sorts of results over the years, nothing to be alarmist about, but certainly, it's not as simple as just going bang, do your test, and it's not a binary issue. In other words, it's not black and white. And certainly, I guess the work going on recently with new technology, some of those will be game changers. I think from our sector, the work that Nikki's doing, the work that Monica's doing, incredibly powerful to be able to survey source blocks, so we can provide a much higher level of confidence in that propagation material to industry. That's really important. And for established vineyards, I guess my last note would be get out there and understand what your status of your vineyard is. A lot of information presented today is a roadmap. I think what Kerry presented with the work that she and Kath have done at Treasury is some really great take-home messages in that, what to do in your vineyard, where to look, and then go from there. And there's plenty of expertise out there. Don't hesitate to ask for advice.
Thank you, Chris. And Kristen's here with us in Coonawarra in the background for any questions or comment. I do have a couple of questions, so I'll put them out first, and then if there are any questions remaining on the floor in either Margaret River or here in Coonawarra, we can go one for one, as we say. So one from Coonawarra and one from Margaret River. So we do have a question, and the first one for Vaughan is without mealybug present, if a rogue vine leaves remnant roots, is there a physical transmission risk? And is there a recommended fallow period? And I sort of felt that this could also sort of angle into Kerry's legacy load comment and the effectiveness of roguing.
Can you just ask the start of that question again? I didn't catch it. If there were no mealybugs left?
Yep. Without mealybug present, if a rogue vine leaves remnant roots, is there a physical transmission risk?
Yeah, without mealybug, no, there probably won't be. It's what I mentioned in my presentation, that it's that potential for overlap between virus and vector where we're likely to see spread of the disease. So, in a situation where you've got remnant roots, I would certainly be looking to try to remove as many of them as you practically can. I know that's difficult, but we're talking for us with the Citrophilus mealybug, we're seeing it to a depth of about 20, 25 centimetres down. So depending on the rootstock, it's actually quite feasible for the vineyard team to be able to remove quite a sizable amount of that root material from that virus-infected vine. You won't get it all. Anything that might run for a few metres or potentially down half a metre or more, don't worry about it. The likelihood of mealybug being down there is probably quite low.
Thank you. And a question for Fiona Constable was, you mentioned that sapon secateurs cannot transfer virus. Can you elaborate on this? Are the virus not viable outside of the host or vector?
I don't know that it's not viable. It's just they need to be deposited quite directly into the phloem. So I think that's more the reason why, and there's never been any evidence of that mechanical transmission beyond that. So really, to get something into the phloem, it's really got to be deposited either by the insect vector or through that connection that the vascular tissues make, then which through it can pass once those connections are made.
Thank you. I'm going to go a little bit off script here because I do have a question from Coonawarra. So I'm going to actually ask Nikki, who's still in the room, to quickly come up. He is looking appropriately surprised. Just with whether technology can detect vines with no visual symptoms, and assuming that you're using vine-only signals, like any thought on that comment made about weed hosts, like could this be detected as well?
What's the last part of the question?
Just any thoughts on weed hosts? There was a comment earlier, and whether this could be detected as well.
It's definitely you can detect the virus from known there's no visual symptoms because if you use hyperspectral, basically it can see what we human beings cannot see. And also you can apply this to different crops, weeds or anything. But another thing, the difficulty, the challenge here is each variety or each other crop or anything, you have to build a new model for this detection because a virus showing different symptoms and in different stage or different varieties or in different combination of viruses showing different symptoms. So it's a lot of work to do in this area. Yeah.
Thank you. And so now I'm going to put it to the floor. Are there any further questions from Margaret River?
Yeah, so we've got one in the room here, so I'll listen to it and then I'll repeat it for you guys, I think.
So, Mike, I just want to know on Vaughan's answer, is there any root-to-root transfer possible of Leprol virus?
Okay, so we have a question here for Vaughan, whether there's a clarification that is there any root-to-root transmission of Leprol virus?
Not that we know of. I would never say never, but we're not aware of root-to-root transmission of Leprol virus. Fiona, have you got a thought on that?
I haven't heard of that for the grapevine viruses, but occasionally it's reported in other crops.
Cool. Any question from Coonawarra? Anyone in Margaret River?
Robin McDermott's got her hand up. She's in the Zoom meeting.
Unfortunately, I can't see that.
I think Robin's unmuted. Are you unmuted, Robin?
I've just got a message from Robin here. She's asking if, was it Mickey, if he validated his infection from his hyperspectral imaging in the laboratory.
Mickey's just coming back up now.
Yeah, of course. We actually have to do quite a lot of testing. It's similar to what Carrie was just talking about. Because like she's doing this visual assessment in the vineyard by walking up and down and also do lots of testing by training themselves, become to a inspector. So is this the same thing as the machine learning method? So we also have to do quite a lot of testing and then to validate, not just to build a model, also validate this model. And the more data we can feed into this machine learning method, the more reliable the result will be. So we can later on even discriminate different viruses or also different diseases. For example, you found a fungi disease or bacteria disease, so you can even discriminate from that as if you get enough data set for this. So this is a huge job. So, okay.
Thanks, Mickey.
Again, any further questions from Margaret River?
Yeah, we've got one. Just hang on a sec.
From the machine learning team, what is the level of accuracy of the virus?
Okay, so we have another one for Mickey. So to the machine learning team, what is the level of accuracy for picking out a particular leaf roll virus?
So in terms of the accuracy, it's not gonna be possible more accurate than the PCR testing, because the PCR testing were the reference for this method. So we can just try to get as close as a PCR testing. So I would say you can't really 100% trust this map that I'm providing, but you can use that as a guideline. At least you get very good idea what is gonna be about, and then you can do more testing by selecting those ones from this map. Something.
Yeah, and I found it interesting. That map of the vineyard presented in I think in the first session, showing where there were the source vines and then the spread throughout the block. I found it'd be interesting to see whether you detected any trends or whether it's possible to see trends over time with that. I think I was speaking to Mickey on one of our breaks here in Coonawarra, and he mentioned that he's similarly clocked up many kilometres, as has Kerry and Kath walking vineyards. So I'll put it one last time to the floor here in Coonawarra, whether we have any questions. Yes?
Wondering, Mike, how far away in the field? I think some of the problem is that we do put together a bulk up testing 5 lines or 6 lines or infection, and they will only go to 1 line in field. How far away is the technology growers? How far away is that maybe serve to provide in field?
So trying to summarize that question, thinking leveraging technology to change the way in which we're sampling or to target the way we're sampling, how far away is the technology from being something that can be deployed by growers in the field? And I think this could be relevant also to what was presented by you, Monica, and the technology there.
Yep, so I'm happy to start with that. We had a bit of discussion around that at lunchtime here in Margaret River. And I think that where the technology sits at the moment for something like the LAMP PCR devices, they are something that with a bit of training, I think a lot of people could utilise pretty well. But when it comes to the sequencing based tests that we're looking at the moment, where they sit at right now, they're pretty fiddly and they definitely require some laboratory support, and the data analysis can be pretty tricky even with the big computers that we have. So I think LAMP at the moment, yes, with some training. The sequencing stuff I think is a few years away probably from being in the hands of growers and agronomists. I know that's the way the world wants to go. How quick we get there, I'm not sure, but I think it could be a while on that one. So.
Thank you. And Mickey, do you have any comment on that as well? How far away may be the technology that growers can do their own flight perhaps?
Yeah, for the simple, like a very obvious symptoms, we can do it right now. There's no question at all. For example, the leaf roll in Pinot Noir or in Shiraz. But the difficult one is the leaf roll in Chardonnay. In those white varieties, I'm still trying to build this algorithm by a lot of testing and a lot of sampling, a lot of flying. So I would say probably in 1 or 2 years time, I will get something very ready to use, something like that.
So the time frame is not too far away. So we probably have time for one more question from Margaret River. Is there any?
Yep, hang on a sec. Reese.
Is there any commentary as to whether there's any differences between infection rates where the warm climate versus cool climate horticulture and the effectiveness between hot climate and cool climate horticulture on things like yield?
What was that last bit? Sorry, on things like?
On things like yield.
Yeah, okay, all right. So let me see if I can summarize this. The question here is whether there's any commentary on the differences between warm and cool climate viticulture and the differences in yield. Yep. And the spread of virus. So if there's a difference between the warm and the cool climates with virus spread and the impact of yield between those two climates.
I might start, but I can't promise I finish. I don't think we've done the R&D in Australia to really understand the difference between climate and impact on yield and quality. I think that's something we firmly need to do, and it's really great that Mon's actually been able to do some of that work already in the Chardonnay, and I think we need a lot more of that because I think it will inform how we manage stuff as well. And I was going to say something else and it's flitted in and out of my brain. On the spread, of course, it's going to depend on how the environment infects the life cycle of the vectors in the vineyard. And again, I think we need more R&D activity on vector transmission of the viruses in Australia, and not just for the leaf rolls, but at GVA, GVB and GPGV, any of the vector borns would be really useful to have a look at transmission risk.
Yes, I think that question reminded me as well of something that I think Jenny mentioned in her presentation, that different seasonal conditions and the movement of vectors. And I don't know if there's any comment I can relay from Jenny or Chris, if you've got comment on that to conclude.
Sure. Yeah, I guess from my observation, both of source blocks and commercial vineyards, there's certainly different expression from year to year. Leaf roll 3 certainly expresses very strongly in my experience in red varieties. GVA, on the other hand, we find is one that varies greatly from variety to variety. Grenache, for example, we do a lot of work with Grenache and Barossa. You see very little impact, either visually or even in yield or grape maturity. I guess it is there's a somewhat unique variety, as probably Mataro, that can tolerate GVA with little impact. And Cabernet Sauvignon will be the same. The old clone 125, as we know it, is well known for GVA, LR1, sometimes 9, sometimes all 3. So it does vary greatly. And I think the cool seasons generally accepted, the replication is potentially a bit higher with the virus. In extremely hot climates, replication tends to be suppressed somewhat, from what I've read anyway.
And I might add to that too, that when we did our trial back in the day, it feels like a long time ago now, we did see differences when it comes to virus and its interaction with the grapevine specifically. The last year, the second last year of the trial, 2009 was when we had that super hot and all the weather and all the bushfires, and it was towards the end of the trial. We could still detect the virus, but at both sites, or all the sites that we had in the Yarra Valley, at Sunraysia and over in Riverland, we did not see symptoms in that year. So that sort of strikes me as a significant impact. And I mean, it would be really great to confirm that, but I feel that that was really an association to environment and temperature impacting symptom expression, even though we were still able to detect virus.
And probably just on the vector side of things, from the work that was done out of Canberra, we know that a cold, wet winter probably kills a majority of scale or a percentage of scale. If it's a cold, dry winter, scale will get through that pretty easily. And in terms of heat, we did think maybe that the scale aren't as prevalent in the Riverland or inland because maybe the heat is killing some of the scale. There's still some work being done around that in terms of the vector and the impact, and I haven't done as much on mealybug around, the conditions. We look at why we have, as I said, this explosion or this increase in vectors, and whether it is a climate shifting in our climate, but certainly weather conditions can play some role in the impact they have on those vectors and how well those vectors survive in a vineyard.
Thank you, Jenny. Now, I'm just going to quickly look to Ollie whether we are running a little bit over time, but we may have a final question in a while. Do we have time, Ollie? Yes. So
in a full removal scenario, what are the using lines for a longer period of time? So competing devices live.
Jenny, I might get you to actually relay that question in your answer.
Okay, so we've had a question from Coonawarra around management of removal of a vineyard, whether we are better to leave the vineyard and treat the vector with a fairly, in Australia, it would be a samurai, in New Zealand, it would be a Confidor type chemical or insecticide, and whether we're better to do that and then rogue out the entire vineyard and replant. So use those plants that are there as a means to control the vector. I think that's the question. Look, the problem I think that there probably is some usefulness in that. The problem we have a little bit in Australia is the acceptability of across the industry of that group 4 insecticides. They're not looked favourably upon within our industry. Even in a rogue out situation, it is difficult. So we are trying to work with industry to say that these products have a particular use at a particular point in our redevelopment strategy. So that's, I think, something we need to get out to industry, that they do have a purpose and we can use them strategically. At the moment, they're really looked at as Europe doesn't like them. So certainly in a productive vineyard, they're not favourably looked on. But I think in a replant or roguing out, we need to use those products a bit more strategically. So, and that would be a good way to do it, is to try and get rid of a lot of the vectors before we replant, even in a grafting situation. You saw that one where we had scale, and we're grafting straight onto that material. We're not going to pick it for a fair while, so there's probably some strategies we can use there with insecticides to do that better. I don't know, Fiona might want to say something. No.
So just with time, we might have to leave it there. And so thank you for all the panelists for staying on, coming back. We did get a message from Dr. Nancy Shallon saying that she couldn't come back, but any questions we can relay via email or provide details there. And so I'll hand back to Uli to close out the day. Thank you.
Well, thanks, Hans. And yeah, it was a great Q&A. And we're a little bit over time, but I think that's fine. How do I end this meeting? Press the button. Thank you to everyone who's been involved, participated in all forms, presenters as well as audience. Thank you to the green room out the back there, My Meeting People and One Vision. And as I said, this is recorded and it will be available to anyone that's interested after the event. It won't be today. It'll be in the next few days. We'll let people know there's been a lot of interest as well. People did actually want to zoom in. We'll look at that for next time. The complexities of this we're just coming to terms with. But I think a successful day. So thank you, everyone. And see you next time.